Are we going to record this?
Oh, yeah.
Oh,
Good
thank you
point.
for letting us, for reminding us. Irene, can you hit the recording?
Yes,
Yeah, I've just started recording, and yes, if you can announce, we can announce that this event is being recorded.
so we can use it for training purposes down the line. Okay, I need to start the presentation to see where the slides are going to land so I can share the right screen with you.
Georgios, before you start, we have Regina from the Department of Agriculture, Florida as well.
Okay, excellent. Now I need to find the WebEx controls and share. Hopefully this will work. Do you see my slide?
It's loading.
Okay.
Not yet. Yes.
Okay. Let me move the WebEx controls out of the way. It's a little bit slow. So is the WebEx controls inside the slide, or you can see the slide clearly?
We can see clearly. Yeah, we don't see the controls.
Well, thank you, everyone. Great participation from around the network. Highly appreciated that you spend the time with us. As you will see, and as Fatima said already a couple of times, this is like it's like step one of a lengthy process. You will see for the CCPP that process started in 2012, exactly 12 years ago, actually, in terms of quality management. So, and since we are essentially a brand new program, since the 2005, where I became the director, we are using this opportunity to, as an educational process, and to see for our team internally, since you are using the CCPP as a case study for the implementation of a quality system in the citrus network of the NCPN. Hopefully you can see the continuum and the history and all the effort that has gone into this. So let's see if the slide is going to move forward by clicking or by page down. There we are. And as always, I put the acknowledgements at the beginning because it's not about the buildings and the budgets and so on. It's always about the people, and we always have a great team, and not only in California and the CCPP, but around the world are collaborators. And of course, the whole network of citrus with the National Clean Plant Network. A little bit of background of the citrus industry in California. Again, for the young members of our team and for those of you that you're not familiar, there is 4 primary cultural zones plus 1 up in the north. We have the desert, the coastal area, inland empire, and the Central Valley, which is the largest. Still, navel oranges are the largest type of citrus grown in California, but the mandarins and mandarin hybrids have been growing constantly for the last few years. In 2017, there was a critical point in the history of citrus production in the US. California, for the first time in history, produced more citrus than Florida. Unfortunately, Florida has been struggling, as you know, with Huanglongbing and a series of hurricanes and so on that they have damaged their crop. Big difference between the citrus production in the United States, especially between Florida and California, is the fresh produced fruit versus fruit produced for juice, for processed. And you can see how California is primarily fresh fruit market versus Florida, that it's a juice market.
Giorgio.
Yes.
Are not advancing for us, for me.
Oh, they are not.
Oh, okay.
How about now? Am I going too fast, or what slide do you see now?
On the first slide, the first slide of the presentation.
And that's true for all of you?
Yeah.
Yes.
Yes.
Okay, let's, it's frozen right now. We hit escape. Anything changed?
No?
No.
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 So you're still seeing the first slide?
Yes.
Yeah. So
Okay, now changing to the,
Word document,
You see a Word document that's on the background.
just the Word document. Yeah.
Okay. Okay, so now it finally clicked something on the WebEx toolbar. Now WebEx is moving a little bit. It's struggling, it's struggling. I'm gonna shut off my camera to help it. And you feel free to do the same now that we're done with the introductions. Okay, so I'm starting again with going to full presentation, switch my screens. To share the right thing, find the WebEx controls and share the right screen. Do you see a slide with a little tractor and a...
It's loading.
Okay,
Yeah.
Yes.
okay. So that was my title. You saw it for a long time. I thank everyone that works with us within the lab, within the US, around the world, especially the NCPN centers. These are the 4 main citrus producing areas in California. As I said, down in the South Desert, Imperial Valley, Ventura, coastal areas, Central Valley, the largest, and a small area in the north where they grow primarily satsumas. Navel oranges, still large acreage, but as I mentioned to you, mandarins are gaining momentum. Let me see if I can remove the controls because they're in front of me. In 2007, are the slides advancing now?
Yeah.
Yes.
Yeah, we can see the chart. Florida, California. Yes.
Excellent. In 2017, as I told you, because of HLB problems and hurricanes, the Florida production was reduced, and for the first time in the history, California produced more citrus than Florida. And this graph shows how, the production between the different states in California in terms of fresh and processed fruit. And California is a fresh fruit market versus processed fruit market in Florida. We are serving a diverse, growing, impactful industry, both in exports and locally. The industry is valued at $3.4 billion. That was a study that Bruce Babcock did a few years ago. He just published another one at the Journal of Citrus Pathology, and he raised that value at $3.6 billion. And there is an estimation that if 20% of the citrus industry is damaged, there is going to be a $500 million impact on California GDP, and over 8,000 jobs lost. So this is what's going on in Florida. Acreage is going down, which is the bars. And even though in the recent years, the acreage has been stabilized, the production, which is the line, keeps going down. And that's exactly what we want to avoid in California. And that's where the CCPP comes into play. We are, of course, as I always say, to keep the citrus industry going is an onion system, right? It's a systems approach. we are at the very outside layer. We are the gatekeepers. We make sure that pathogens do not enter our propagation pipeline, because after that, any other program trying managing a disease or a pest is doomed to fail. So by law, that was instated in the 1950s by federal law, citrus cannot enter the United States unless if it comes through a program like the CCPP that operates under a specific permit. And a few years ago, that's why I have the little FBI icon down there. I always tell to my audiences that there is always someone watching. So there was one day, and Robert was with me, special agents from the FBI agents from the San Francisco branch that they were monitoring industrial espionage cases. They came and talked to us about how we handle the citrus badwood and who do we send it to and so on. So we're operating at a very high level. The federal government is trusting us, and we make sure we're always, follow the rules. And that's why the quality is very important in our case. We cannot miss once. We cannot have the wrong test result or, make a mistake. The consequences will be severe. So as I told you, the CCPP operates at the first layer of keeping pests away. It's the most cheap and efficient way to deal with the disease in general as a plant pathology principle. When the disease escapes the exclusion measure, you have to attack it and eradicate it. That's where we are today in California with the ACP, with the Asian citrus psyllid and the Huanglongbing. But you see how from CCPP costing roughly $1.5 million a year and keeping away all known diseases, we spend over $40 million a year just chasing one insect. So it becomes super expensive and difficult. And of course, if the disease escapes the eradication level and you have to manage it, you have to live with the disease, that's where the cost becomes in the billions and the economic losses become tremendous. That's why our program, as I told you earlier, cannot miss once. And we are operating on the very basic principles of plant pathology. The classical disease triangle. We cannot control the environment, but we can control the pathogen. We make sure we remove the pathogens from introduced varieties, and we make sure our propagating materials are not getting infected with diseases. And we make our hosts that now they have been treated for diseases available to everybody that needs to propagate citrus, either in California or in the whole United States through the NCPN citrus. And why this is important, why availability of pathogen tested host is important. This is a very crude history of evolutionary history of citrus practices, I would say almost globally. So in the early years, cirrhosis in the 1800s, early 1900s, cirrhosis was a big problem. And the solution that researchers found primarily here in the citrus experiment stations, some of you maybe have seen varieties called Frost Nuceller Lemon or Frost Nuceller Navel and so on. Growing seedlings, they were free of cirrhosis because most of the citrus pathogens are not seed transmitted. But seedlings had issues with fruit quality and Phytophthora, so the industry switched to sour orange rootstock. That happened. There is propagation records early on, so sour orange was dominating all the way to the 1900s. But then in the early 1930s, Tristeza quick decline hit the industry and killed 100 million trees that they were propagated on sour orange rootstock. So the industry shifted to the trifoliate and trifoliate hybrid rootstocks. But these guys, as a lot of you know, they are sensitive to viroids and more severely to cytotoxocortis viroid. And these are recorded turns that the industry took on its evolution, and who knows what kind of turn is going to take in the future when we find the HLB tolerant rootstock or the HLB tolerant scion. So if imagine in every of those yellow arrows when you make a turn and millions of trees are propagated on this new species, if there is not a clean source for those species that you're trying to replant your industry, you are exchanging one problem for the other. And this is happening real time. For example, in my home country in Greece, Tristeza was recently introduced, the switch to trifoliate hybrids. There was a publication actually from our lab that viroids are prevalent in Greece. They had to rush because they were losing their orchards, and now they're having exocortis and so on problems on the trifoliate rootstocks. So it's very, very important when you're making massive turns like that to have access to pathogen-tested propagated materials. And in our case, we're the first in the world that realized that, thanks to the discovery from Dr. Fawcett in the 1930s, that figured out that if he goes to a psoriasis tree that you see there on the left, on the right, the bark scaling symptoms, if someone goes in a psoriasis tree, symptomatic tree, and propagates trees, all the trees will have psoriasis symptoms. So they started back in the 30s, the psoriasis freedom program. And parenthesis here, you can see the original faculty of the station sitting on the steps on the back door of our office at Mount Rubidoux, where the original experiment station was. And I don't know if you can see my mouse moving, but young Dr. Fawcett is right here. And these are the guys that are buildings now in our campus. This is Boyce, Weber, and I believe Dr. Frost, it should be this gentleman over here that we mentioned earlier. So in California, we have been excluding pathogens from our program for decades now. And pretty soon it's going to be 100 years that we have been doing it. And you can see how the program shifts and evolves almost every 20 years as new technologies are emerging and so on. And we are in a transition right now with the next generation sequencing and the National Clean Plant Network. The recent establishment of an endowment for the sustainability of the CCPP. So a key on this rough history of our program here, a key point was CCPP joining the citrus network of the NCPN. So NCPN was established with the Farm Bill of the 2008, and it has been renewed ever since with the Farm Bills, and most recently became a permanent program within USDA APHIS. And the key, what made the NCPN different in terms of legislation was that it covered existing programs. You couldn't start all of a sudden say, oh, I'm making clean plants of this species and I want to join. You have to be doing this job already. And you had to do three very specific things. You have to look for diseases, diagnostics, you have to perform therapy to remove pathogens, and you have to maintain foundation blocks for distribution of your materials. And again, you see how diagnostics, and that's why we start from that point, and you will see more evidence later why we're doing it. We want, the quality to be implemented at the CCPP, starting the diagnostics. The NCPN has spread to several different commodities, and it has different centers throughout the United States. So I want you to remember this slide. This is the slide that's going to come up towards the end of the presentation to connect us, because now I'm going to transition to what actually CCPP does to connect us to the quality initiative of the NCPN. So programs like CCPP survive because they're collaborative programs, and there is scientists, there is regulators, there is industry involvement. And true to the NCPN mission, we import material, we perform diagnostics, we perform therapy, we maintain the the foundation materials, and we distribute our disease-tested, pathogen-tested propagative materials. We are achieving this by doing these very basic 6 things. Bring material in, we check it for diseases, we perform therapy, we have the foundation materials that we test all the time, we distribute, and of course we perform extension outreach and research. I found this board slide is actually from my job interview, where I tried to capture the program in one slide. Had some cool animation back then, but there is no point of doing this today. And it's still true today. Some numbers are not accurate, the 18 months limit in the nursery block and so on. But this is primarily how our program indicates. Left to the red line is the quarantine step, and the material cannot cross the red line, this yellow arrow here, until it has finishes the variety index and moves to the foundation facility for distribution to the industry. And a simpler schematic of our pipeline is this. Material comes in, we perform what we call pre-indexing or initial index. This arrow going directly to variety index has been eliminated since the new regulations took effect. For Huanglongbing, everything needs to go to therapy. And after we have evidence that the therapy was successful with laboratory testing, then we proceed to the variety index, which includes both biological and laboratory tests. And when variety index is successful, and the variety receives its VI number, then it's released to the foundation facility. Fatima, how am I doing with time? I know we're a little bit late, but give me a time so I will know how fast I need to go.
You're supposed to be done 9:50.
Okay, I have 20
We
more
have
minutes.
12
Okay,
minutes, yeah.
thank you.
Mhm.
So the CCPP today has 12 people, and it operates on 3 different facilities, and a budget, as I told you earlier, of roughly $1.5 million for core activities. And this is how a structure looks like. And from the people from my team, you can see more or less where you fit, but we have our administrative unit, we have our production unit at Rubidoux, a production unit at Linkov, and we have a laboratory unit for the tissue culture lab and the diagnostics lab. And just to give you a perspective, I'm going to try to do throughout the presentation again, because we're a brand new team. This is how the CCPP looked like in 2005. And our budget back then was roughly $200,000. So there was no administration unit. There was a couple of people at Rubidoux and 1 person in the lab. Just to give you an idea, Southern California at the Riverside, the city of Riverside, we have a Rubidoux quarantine facility, and 4 hours roughly more north, 240 something miles north, in the San Joaquin Valley, we have our foundation facility at the Linkov Research and Extension Center. So what's going on at the quarantine facilities? We have our laboratory, the research therapy and diagnostic laboratory in the main campus, and we have the Rubidoux quarantine facility at the site of the original citrus experiment station, which you can see on the back there of the image behind the glass houses. So this is what I inherited in 2005, and the facilities that they were there were from the 1950s. And most of you have not seen any of this. And this is the beginning of the new generation of people coming to the CCPP to bring it to the 21st century. Our head house was completely dysfunctional and built with transite, so we had to have an abatement company removing the carcinogenic elements. We ripped apart that old building that wasn't offering anything. And we slowly start building the foundation and what all of you know today as the Delfino Family Plant Laboratory. We name it to honor one of our private donors, the family of one of the private donors, Tom Delfino. This is our screen house, again, built in the 1950s. And of course, there was no way that we will pass. Remember now, this is 2005. The HLB epidemic is taking off in Florida. There was no way that this facility will pass any kind of inspection or protect us from Asian citrus psyllid or anything like that. So it was a beautiful structure, but unfortunately we had to take it down and slowly start replacing it with the stainless steel structure that most of you know today. That happened roughly in 2013, between 2011 and 13. This is how the glass house used to be. This was in one of the original glass houses, aluminum glass houses in the state of California. And now we have built these vestibules where they're supporting our needs, backup generators, steam generators. None of this existed when we started. Outdated cooling systems that we upgraded and we protected with screens for the psyllid. Every few months, we had to treat the wooden benches with copper naphthenate, which I don't know how many of you have worked with it, but it's not a pleasant chemical to work with, so we replaced with benches. And we got rid of these dilapidated trailer buildings that they were given to us, that we inherited as office and laboratory space, and we moved to the original Citrus Experiment Station building, and most of you know this as office space and conference room. This is the equipment, Paulina, that you were supposed to do SOTIP grafting those days. A wooden box with a fluorescent light and a super old laminated laminar flow, and this shade cloth was the was our SOTIP grafting incubator to transfer plants from the wooden box to another wooden box. And of course, today most of you have seen that we have proper equipment to do tissue culture as far as SOTIP grafting going. And of course, Paulina is working to set up a proper tissue culture lab for us with media room and so on and so on. So this slide is from a presentation I gave to the dean that we took care the big old stuff in 2011-13, and then we start fixing other things like our water pressure, which we couldn't install any automatic systems before that. We were on DSL up to very recent years. And as you're experiencing, all of you at Ruby right now, we're halfway through of fixing items 3 and 4, bring more power and take care of our greenhouses and so on. And of course, there is a long way to go in the future. My time is running out. Most of you know about the SOTIP grafting process, so I'm not going to play the video, but essentially we excise a small piece, smaller than 2.2 millimeters, as you can see on the right. And we are repropagating a tree in vitro. And after a few weeks in vitro, we're pulling out the little seedling from the tube, and we propagate it on a rough lemon rootstock to grow fast. And we transfer our babies in the growth chamber for perfect growing conditions. And eventually you end up with a tree like this. You have the rough lemon rootstock at the bottom. You have this lighter color here, which is the Carrizo interstock that was the rootstock on the tube. And then you have the variety growing on the top. Now, because you did the therapy, and that's how a tree looks like, because you have done therapy, that doesn't mean the therapy was successful. So for us at RubyDo, this is when you start doing what we call the IPPN care, and you find those sources and you put them to the test. You put them through laboratory tests and a series of biological tests. And this is a few early upgrades that we did in the lab in the campus. A brand new microscope, high throughput extraction equipment, qPCR machines, and so on. And by doing this process, we're not, as I told you earlier, we're not excluding only one pathogen like HLB, but several different pathogens that they cause millions and millions of dollars in losses in other countries. So because of those upgrades, you see here in 2013, we got our first set of innovations. You can see how material coming into our program has been growing tremendously. you can see how our therapy capacity has increased tremendously, especially after we got the specialist in tissue culture coming to our program. We have also been increasing our output releases from quarantine. The main bottleneck here has been greenhouse space, but with the conversion of our greenhouse to a greenhouse, now we have a little bit more space, and we hope to increase even more our output. And where we're focusing right now is to compress the time that takes us to complete the process. And what this slide is showing you is that when you start with OB, with original budwood, right now we're averaging, I haven't added some recent varieties, but we're averaging a little bit below 30 months, 2.5 years. But if the original budwood fails and you have to use greenhouse sources, then the process becomes very lengthy, over 40 or close to 40 months. So receiving budwood at good shape is very, very important to us. So there was a variety that was giving us trouble, and we came up with this idea: what if we put temperature sensors in the shipment? And we collected some data, and we saw that actually the budwood can reach very high temperatures, and this specific variety came coming cooked to us, as you can see here, the brown bud stick. So we approached USDA and we amended our permit to be able to ship budwood in culture. So we have, as I told you earlier, always the program is evolving and try to address issues. So budwood in culture in media arrived in very good shape, and actually by the time it arrived, it had already flushed, and we could do suture grafting immediately. And we have been facing always problems, but we're always finding solutions. And what this slide is telling you is the blue circles and the boxes on the top, the different stages, 1, 2, 3, shows you how us making investments and how the industry making investment has been addressing these issues for the CCPP. The blue dots are months in quarantine of different varieties. And you can see how moving forward from 2005, how those blue dots have started clustering. They have been compressed. They're not all over the graph like the before 2005. And they're all tending to be towards the green zone between 24 and 30 months in quarantine. And not only that, this graph is showing you the orange line is our capacity to bring in new varieties, and so you can see how we have increased our capacity tremendously. So when you start looking at those blue bars, those blue dots, and looking where our time is invested, you can see that therapy, the first steps, where is the blue part of the bar, is very compressed. Then we are losing time trying to grow plants, which is the orange part of the bar, and we're losing time on the gray bar, which is us doing testing. So the two areas that we really have to improve is how we grow plants and how we do testing. And again, that's the reason for the quality. That's the reason on the research on diagnostics and so on. And this is a little bit more detail of some more recent variety releases, and the 104 weeks is where we want to be, 2 years. And you can see how we keep compressing our introductions towards that, but what is keep pushing us out from that range is the lengthy growth, which is the gray bar, and the indexing part, both in the lab and in the greenhouse. Okay, it's 9:50. I'm going to steal 5 more minutes to bring this to a wrap, because I'm pretty sure we started late. So after we finish our VI index, the varieties we said released from quarantine, and it's moved to the foundation block or the foundation facility. And a little bit of history of the facility started in 1961. This is Chester Roystacker planting the first tree in the foundation block at Lindkov. And of course, since then, a lot of investment from the industry and the university. In 1998, we built the first greenhouse because we were anticipating Tristeza to be the big problem of the future. The screen house was expanded in 1999. In 2010, we built a second screen house, and in 11, we built the positive pressure greenhouse so we can have duplications and overlaps of our badwood sources so we will not lose any trees. There's a few pictures from the construction of 2010 and a few pictures of the construction of 2011 for the positive pressure greenhouse. And the system works. We had incidences where they gave us faulty materials for the screen, and the roof of our screen house opened from one end to another. Rob has been taking these pictures where birds have been pulling threads out of the screen to make nests. So all sorts of things happen to facilities. That's why it's important to have overlaps and duplications. When we encountered this problem with this was a faulty thread that they had used to make stitches. So we approached the companies and we said, we cannot afford this. No more stitches. So they engineer for us these metal clips that they keep mechanically the screen attached to the frame. And I don't know if you remember the slide where there were only 4 people at CCPP. At that point, there was nobody permanently at the Lincoln Foundation facility. But since 2012, we have Rock, and a few years later, in 2016, we got an assistant up there, and improvements were made in the fertigation. Plants, fertilized properly. There is all sorts of countermeasures for frost protection, repropagation of all the badwood sources in the positive pressure house for safekeeping, and digitalization on the foundation block, which again goes to the quality quest, and digitalization, as you will see later, at the laboratory. And this digitalization is what has allowed now the Linkov Foundation facility to process orders electronically all the way through, from someone placing an order in the online system all the way of the badwood system. Trueness to type evaluation, as you know, is very important for programs like ours. The old way of doing things, as you can see here on the top picture, static websites and seasonal testing and so on, has been going now out of the way, and we are now have engaged in a discussion with the industry how it's not only about fruit quality and so on, is how we can build a holistic post-quarantine management system, and we are engaging other citrus programs at the university. So really, we can take care of all steps, make sure that the badwood source is producing is distributed badwood of the right type, and make sure that the horticultural properties of a variety have been evaluated in the long term. And of course, in the heart of the citrus industry, we always have outreach events, and we display our fruits. This is, and the fruits from our collection. This is the first HLB tree in Los Angeles. It was discovered in 2012, and that tree had 23 grafts on it, and nobody knew where the badwood came from. So that was a eureka moment for the industry and ourselves, that there is thousands of people out there propagating citrus and nobody knows what they're doing. So by 2012, we had the people in place, we had the electronic systems in place, we had the facilities in place. So we opened our program. The program was dedicated, because it was sponsored primarily by the citrus industry, was dedicated to service the industry. But at that day, at that point, the industry made the decision we need to open it to everybody because this happened in Los Angeles. And you can see the staggering amount of people, which is the bars, that they are ordering badwood from the CCPP. And you can also see the tremendous increase in volume of badwood distributed. And in the parentheses on the bar where the years are, you can see specific, turning points. In 2014, the Fruit Mentor website appeared. And now the public has a very popular platform. Fruit Mentor YouTube channel has millions of views and 500,000 subscribers. It competes with like rock stars. And that's a big reason why we think the hobbyists are, coming to CCPP for their budwood. And throughout the years, the original price budwood was set in 1984, and it was 75 cents. So we have been revisiting the cost of budwood, and late 2021, price of budwood increased for the non-industry members to $5. The reason that the industry members keep paying the lower price is because they are assessed, they are taxing themselves for their citrus production, and this money is returning to CCPP in the form of support from the Citrus Research Board. And this is how a budwood cut looks like, and we have been using, because we now have access to all these thousands of people, we're using the budwood shipments also to distribute educational materials as needed. And what I have been describing to you what is happening in the physical world, but there is tremendous innovation also in the online presence, in the digital world for the CCPP. And all of you know the tremendous work that Peter is doing both on the citrus variety introduction system and the budwood distribution system, and all our efforts to bring this together under a LIMS program, a laboratory management, laboratory information management system, so we can finally meet the, one of the basic requirements of a quality management system. So to recap, the CCPP has been growing because a tremendous investment was made on us, and we have been very successful of moving the program forward. And we have been attracting attention. This is the first time in the history that the Chancellor of UCR visited CCPP. And here is me lecturing Secretary Ross, Secretary of Agriculture in California, Karen Ross, about the upgrades we have been making and looking at the suited grafted plant over here and so on. So we are at the forefront of developments. We have the system is trusting us, they're investing on us, and we have been attracting attention both from the public and from regulators and administrators. Now we're out of time, so we don't have time to go through all the research we're engaged to, so we can keep moving our technology forward. So I'm going to skip through all the individual slides really quickly. We may stop only for 1 second, maybe in a few. We have been working since the 1800s with the city of Riverside to keep a parent navel tree alive and protected. A lot of you know about the effort we made to make sure we get Florida breeding programs materials into California. Work from Fatima and Douglas on consolidate our diagnostics and engineer instruments that they can do great work for us and cut down the time for sample processing. Also a great project with Oklahoma State University to bring high throughput sequencing technologies as a diagnostic tool, and work with CAFE on using modern indoors production systems so we can cut down the time to grow plants. Again, we don't have time to see a full video here of how the test team had built these budwood processing tissue instruments for us, but everything is digitalized. We can scan a tag over here and it uploads the information of a sample in the cloud, and then just in a few seconds, what it used to take us 24 or more hours, now in less than a minute, as you can see here from the video.
Plant material. And then this syringe, which has captured all the plant material, it has some guanidine in it and a filter.
Let me see if I
to
can mute
separate
the
plant
video.
material.
As you can see here, in less than a minute, we have crude extracts that they can go directly for nucleic acid extraction versus overnight freeze drying steps and grinding individual tubes and so on. We're trying to assist the industry with high density plantings and dwarfed citrus. And you heard earlier from Dr. Pagliazza about how we're trying to bring into the CCPP sustainability principles and circular economy in citrus. Yes, we're at the beginning of the pipeline producing trees, and we want to make sure that we come full circle, use citrus waste and bring it back into the CCPP to expedite citrus growth. And you can see here a case where adding biochar in this graph here on the right, adding biochar produced in this case from almond cells, another agricultural byproduct, has increased germination of seedlings for the CCPP, which it's the red bar versus the control, the blue bar. We are engaged in studying citrus phytobiome because citrus are not sterile plants, and we need to make sure we know what's going on internally so we can promote them. We're helping breeders and researchers with HLB tolerant varieties. We have been helping Hai-Ling Jean in her projects with, she identified citrus occurring small peptides for management of HLB symptoms. Wembo Ma with her projects on secreted proteins of the Candidatus liberibacter. And James Borneman, where he's building metabolomic models about the 3 organisms involved in the HLB pathosystem, so we can identify targets for disease management. We've also accommodated the K9 training and deployment in California, and the discovery we made our lab with the citrus yellow vein associated RNA to be used as an expression vector for disease management. We have been working with USDA ARS on cryopreserving varieties. The USDA repository for citrus and dates has taken the lead on these projects, and we're there to support with plant varieties as needed. And of course, the pride and joy of our program, the high throughput pipeline that has resulted in a tremendous impact on all sorts of diagnostics programs in the state of California, and of course had the greatest impact at the implementation of the Clean Nursery program in 2010, for which you can see in the red bars, we increase the capacity of the program in thousands of samples by qPCR, and we have brought down the black line here, we have brought down the viroid incidence in citrus nurseries. And we received in 2018 the American Phytopathological Society Award for Excellence in Regulatory Affairs and Crop Protection because of this system. So to transition to the quality management, when in 2012 the CCPP went through an external review, it was recommended, but essentially became an industry mandate that we need to implement a quality management system at all the functions of CCPP. And the quality management system is as simple as this. Nobody asks you to reinvent the wheel. It's all about the things you are doing already. And all you have to do is to say what you do what you say, prove it and improve it. And that's what we have been doing all these years. We are trying to implement the different elements of the quality management in our program. And today we start with this training. We have taken a lot of steps, which you will hear later, but today we're focusing on the testing methods. And quality is only one of the few initiatives of the National Clean Plant Network. There is initiatives on economics and education. And because no quality management system is one size fits all, we thought what the quality management system that works in California is not in the California Citrus Center is not going to work in Hawaii Citrus Center or in Alabama Citrus Center. So we took the initiative, because we're one of the largest centers in the network, to start this process, have a case study for CCPP, and hopefully we'll be able to move forward with implementation of quality management systems throughout the citrus centers of the network and other citrus diagnostic laboratories. I went over time, but I think it was important for all of you to see the full picture. I can stop sharing. I don't know if you have any questions, but we will be together for a long time. You can ask questions at any time. I believe next is, because my computer is slow, next is the introduction to the quality initiative, correct?
Correct.
Thank you, Bruce. Yes, it is.
Thank you.
Yeah, I don't know if while we're loading the presentation, anybody has a quick question for me. But again, I hope I gave you where we have been, where we are, and where we need to go, and how all your hard work and teamwork and collaboration between all the different agencies really have brought up our program at a completely different level. All we need to do is now to wrap it up and bring the quality management principles in so they can carry us sustainably forward. Thank you, Irene.
Thank you. Okay, then I will start with this presentation. Good morning to everyone. For those who joined later, I'm Irene Lavaji-Kradek, and together with Fatima Osman, we lead the National Clean Plant Network Quality Initiative. And today's workshop is really a treasured opportunity for us to tell you where quality in your everyday work fits within the larger framework of the National Clean Plant Network efforts. So I'll tell you how we got here today, the state of the art, and where the initiative is going, that is towards the creation of a National Clean Plant Network system-wide quality program. So first, let me give you a brief summary of what has been done so far for the National Clean Plant Network Quality Initiative. And we should start perhaps with how this initiative came about. That is, the idea of creating the quality initiative began because NCPN recognized that there is a need to approach and advance quality at the conceptual level within NCPN. And now, in order to do that, as Georgios alluded to, we need to create a clear understanding of what quality can and cannot do for the various levels of the NCPN centers. So quality in the NCPN program is seen as part of the core diagnostics, therapeutics, and foundations missions of the NCPN centers, processes, and products. But quality is also a manifestation of the network overall, of how we share information and the various NCPN administrative activities we're involved with. The NCPN centers, we must not forget that they represent 7 commodities that are very diverse and different in their capacity. And so we need to avoid the one-size-fits-all approach and create a common quality definition, a quality plan that maintains the identity of NCPN, fits its mission, and establishes its needs and expectations at the local and network level. So really a quality plan that aims to create a clear understanding of what quality can and cannot do for these various levels of NCPN centers in over 30 different locations. How did we get here today? Well, the wide-ranging quality concept that was articulated in the National Clean Plant Network Strategic Plan gave rise really to the creation of this NCPN Quality Initiative, which then was developed over the years and supported by various grants. In the first year, there was a first NCPN-supported workshop that took place in 2019 in Riverside to engage NCPN center directors, managers, and stakeholders in an analysis of what quality means within NCPN. There were talks, hands-on activities that introduced the participants to key concepts and principles of quality management, and also looked at existing accreditation programs. And this was done in collaboration with the USDA and the National Plant Diagnostic Network personnel. And then at the end of the workshop, a smaller group stayed behind to draft a roadmap of what the next steps may be, which led directly to the second year of the initiative. In the second year of the initiative, we formed an NCPN Quality Steering Committee. A working group drafted NCPN-centric standards. And there were 2 training workshops in collaboration with, again, USDA and the National Plant Diagnostics Network for QMS and auditor training. And then finally in June 2020, right in the middle of COVID, a webinar took place to introduce NCPN audience to NCPN like the quality concepts. So zooming in into each of these objectives, the steering committee, the members represent different commodities, larger and smaller centers, with the idea of involving, really involving the entire network. The committee is primarily tasked with developing a prioritized plan to best leverage and coordinate NCPN efforts to enhance the adoption of clean plants, and they meet regularly over remote working platforms to review documentation and decide on the next course of action for the initiative. Another activity of the second year was the establishment of a working group to draft national standards for NCPN quality. And this ad hoc group was established as a result of the first workshop and included members who expressed an interest in quality and represented, again, larger and smaller centers and different commodities. The group met regularly via remote working platforms, and they reviewed and evaluated the existing already available standards with the view to develop standards that are applicable to NCPN. But really, most importantly here, the work was to include not just the diagnostics, but also the therapy and the distribution, because these are key elements that are specific to NCPN. So the group made significant strides, and the NCPN steering committee is now reviewing the draft of these standards, with the view that the NCPN standards should be voluntary, scalable, attainable, and really not focused on the mere compliance, but really on the production of a quality product. It's not just about ticking a box for the sake of ticking a box and say that you've done something, but it's ticking a box if it makes sense and it allows you to produce a product of high quality in a timely manner to be able to give it to the industry. And so with all these activities, the group was left to ponder on a series of questions, such as, is quality an individual effort or is it a team effort? What have our centers done so far in terms of quality? Have they already implemented quality strategies? And do we need a plan, a roadmap to help direct us?
So we do need a plan. The answer is we do need a plan. So what does a plan look like, and who will decide what its component will be? So we wrote a proposal to seek funding from the NCPN Governing Board to develop a system-wide quality plan that was crafted with the guidance of a core quality steering committee and some associated ad hoc quality teams to help plan and initiate the activities. So the core permanent quality team was tasked with developing the NCPN quality plan, was composed of 5 to 6 members, the NCPN management and quality initiative leads and quality managers in each of these respective NCPN centers, or even person currently supported by NCPN in some capacity as quality managers. So one of the first recommendations of this core quality steering committee is to survey all NCPN centers to determine what the level of quality is in different centers, their needs and benefits of adopting quality at their own level. So this survey will allow the quality plan to be more in touch with the realities and needs of the people it serves. So we did actually have the survey, and we collected feedback from all NCPN centers to assess their interest in different aspects, such as harmonization of the NCPN protocols and procedures, the need to create an NCPN-centric standards that, as Irene mentioned, it will not be a requirement, but just it's going to be aspirational, allowing the centers to choose some components of the standards and what they need to implement at their level. One of the major components of the plan also is conducting quality training program as per the needs of the center in the 3 missions of NCPN: diagnostics, therapeutics, and foundation. It also offers voluntary program reviews and possibly also collaboration with outside entities where there's a common ground for quality between all the centers. Next slide, Irene, please. So this slide shows you very, very briefly the outline of our survey. We created the survey, we created the criteria in diagnostics, therapy, and foundation. We used SurveyMonkey. We did, among us as the core permanent team, a mock trial to make sure that the survey is comprehensible. We incorporated some comments, and then we sent the survey to centers directors back in June. And we also helped them out with a Zoom call just in case they had any questions. And then we collected the survey results and feedback. Next slide. So the main, So the way we presented the proposal this year, it's okay. Next slide. The way we presented the proposal this year to seek the NCPN funding was by determining certain milestones and timeline to show when and how each of our objectives will be achieved. So last year we have crafted the quality plan with the guidance of the NCPN core permanent team. This quality plan will be, in the future years, will constantly be adjusted as per the survey results and the need of the diverse NCPN centers. In year number 3, the plan again will be fine-tuned and updated and revised after each cycle. We also, based on the survey feedback from the centers, different level of needs were also identified throughout the entire network and were even ranked from low, medium to high. Therefore, the second objective was to create an ad hoc working team tasked with different aspects of quality. The first ad hoc team that was recommended by the survey was to collect and review existing protocols and procedure. Another ad hoc team is to assess the quality training. The third one was the NCPN standard review. The fourth objective of NCPN standards that Irene mentioned is we currently are completing the review of the overarching NCPN standards. And then there is an idea also in future years to create commodity-specific working group. As a matter of fact, there are 2 groups within the 7 NCPN commodities that are starting to create their own crop-specific manuals. Next slide. Another objective, as I mentioned, is the harmonize of NCPN protocols and procedure. And we did talk about this in our Tier 2 meeting, and we are starting to collect and review protocols and procedure. And this year, we're going to only focus on diagnostics and therapy. Second year, we also collect other protocols in foundation, in governance, in distribution, and then share these protocols. The aim of this is to share the protocols and procedure network wide. One of the 6th objective is a training on quality aspect, which we're currently doing right now. We're creating tailored grassroots training programs for different size centers. One of which is the qPCR right now, because the survey mentioned that qPCR is one of the things that is needed together with high throughput sequencing. We're going to continue creating tailored quality training in high throughput sequencing, shoot tip grafting therapy, and maybe even in administration and the collection in the proposal writing and stuff. The 7th objective is conducting voluntary NCPN program quality reviews. This will not happen this year, but it will happen next year. We're creating also an ad hoc working team to start working on the criteria for the reviews. Next slide.
Okay, so you heard from Fatima about the survey. And so based on the results obtained from surveying the NCPN centers, 2 main areas of focus of quality were identified. First, the collection and review of protocols and procedures, and the training in different areas. And so today's training, just to emphasize again, that falls under really the umbrella of the quality initiative training. And this is just one aspect of it. So here is this year's timeline showing you the activities for NCPN quality that we are performing. And one of the main goals for this year, the collection of protocols and procedures, is happening now. Between now and May, we're collecting the protocols for diagnostics and therapy. and representatives from each commodities have been identified and are involved, and they're facilitating the task. But another goal that we're working towards is the training. And the committee, the NCPN Quality Steering Committee, identified qPCR as an excellent candidate diagnostic method to run a first NCPN-wide training. And so today is an excellent opportunity because it's a case study on how we can run this type of training. As George has mentioned earlier, this is we're doing for the CCPP, and we are expanding to the other citrus centers, but with the view eventually to expand it to the rest of the network and include the other commodities. Bringing it back now to the quality of the citrus clonal protection program, here's an example of the quality program implemented at the citrus clonal protection program. So quality, as you've seen extensively through George's presentation, is not new, is nothing new, and is really seen as a continuum of the CCPP, providing the glue between the operations leading to the timely release of a quality product to the industry, the clean buzzword. How do we achieve this? At the level of diagnostics is by consolidating our singleplex into multiplex assays that are already validated and implemented. We're implementing high throughput sequencing in our diagnostic pipeline. And we're establishing LIMS, which we are in the process of connecting to our qPCR machines, just to keep it relevant to today's training. At the level of diagnostics, therapy, and distribution, we're improving the way we grow plants. As an example, here you have a picture of hydroponics for bioindexing. And as George has mentioned, there are other research efforts and activities to improve plant production in general. We're implementing automated systems, and this is for example, irrigation, for precision agriculture, and through the extensive use of databases to follow the varieties throughout our pipeline, from introduction, therapy, diagnostics, distribution, but also very importantly to record the communications, the feedback from customers, and really the movement of the material after it exits quarantine too. And finally, it all comes together under the umbrella of a quality management system, which is organized at present as both files in a Google Drive and also as a quality management system LIMS module. So clearly, quality appears in many, many different aspects of the activities of the citrus clonal protection program. But today's training will focus, as you know, today's and tomorrow's, on this portion of the diagnostics and specifically the qPCRs. And with this, I thank you for your attention.
Thank you, everyone. I'm looking at the agenda, Fatima, and where is our break? We are 10 minutes behind, or almost 15. So I don't know if anybody has any questions for any of this. Okay, so again, it's not only you grabbing the pipettes or the grafting knights do bioindexing, all the pipettes to set up PCR reactions. Everything we do belongs in this greater scheme of things. The evolution of our program that, as we said, in a few years will be 100 years old, and the constant incorporation of modern principles and ideas to our programs. If we don't evolve, organizations don't grow, and organizations that do not grow, they decline. And that's also the beauty and why all of our programs are associated with the university where the research is. So we're very excited that we bring this concept. We have been trying to implement this concept in our program, the quality management concept. And now you can actually see it materializing in several different areas, and all of you play an important role in this. And unless if you have any questions, we can take our break. This is where we're going to transition to the specifics of the qPCR. So feel free, if you are not interested in the nitty-gritty details of how a qPCR reaction works and how you can troubleshoot it and so on, take your break and come and join us anytime you want if you want. That was what I was planning to say at this point, to help you to summarize the big picture and inform you where we're going next. Anything from you, Fatima or Irene?
I think we should take a 10 minutes break and come back maybe 8.
I will do the timer on the screen. Is
yes.
very useful. I'll put a timer right now for, I'll sit a little bit. I'll make it 8 minutes so I can bring you back in time a little bit.
Okay.
Can you stop sharing?
Can you guys see my screen?
Yes.
See?
Perfect.
And also, if you just, down on your bar on the WebEx bar, there is reactions. If you want to give us a thumbs up or a raise hand to know that you're back, or turn on your camera for a second. Okay, yeah, I see some activity. Cool. You guys are experts. Okay. (10:39), Fatima and Irene. You
Yeah, should you just give it 1 minute more and then we start?
Sounds good.
I saw enough
Hands up.
activity in the participants. Yeah, so start loading,
Okay, then we
start
start.
sharing. And Irene, I don't know if you restarted the recording.
Yes, I did.
Yeah, good.
Thank you.
Okay.
Okay. So hello, everybody. My name is Fatima Osman. I am, I guess I introduced myself already, but I helped a lot in designing a lot of the qPCR assays at the CCPP. And my mission today is just to give you a brief overview on the principle of qPCR, which, and afterwards, Samantha Barnum of the Real-Time PCR Research and Diagnostic Core Facilities will take over and go into details on each and every aspect of qPCR. So what is the diagnostic PCR? What is the quantitative real-time PCR? It's a diagnostic technique that is based on the detection of fluorescence produced as a result of the amplification of a target sequence. In contrast to traditional PCR or conventional PCR, where the presence or absence of a DNA sequence is analyzed on a gel upon the reaction completion or at the end point of the reaction, the results are viewed on a gel. qPCR monitors the DNA amplification in real time as the reaction progresses. The qPCR detection is based on changes in the fluorescence, which are proportional to the increase of your target sequence you're trying to detect. It does not only detect the nucleic acid in the sample, but it also quantifies the concentration of the nucleic acid using the fluorescent dye. So as the number of the gene copies increases during the action, so does the fluorescence. So those are the different real-time PCR platform. I thought it was a good idea to familiarize ourselves a little bit with the machines out there. There are several real-time platforms in the market that supports real-time qPCR. This is the list of the top player in the market. There are different qPCR platforms, and the way that are different is they differ in the probe chemistry it supports, the range of the dye, fluorescence, wavelength it can detect. It also differs in the sophistication of the detection software and sample format, whether they take 96, 384 wells, or even other formats. Those are among the earlier models. The Smart Cycle from CIFID, it can only accommodate 16 samples. Then other platforms have evolved to accommodate 32 samples, and all of the other formats are they can detect 96 samples in a 96 wells. Those are the qPCR platforms that run digital qPCR, and currently in the market, there's also on the right-hand side, the MIC or real-time PCR system. Those are mini size that can be brought in the field, and results can even be seen on your own smartphone. Those are the widely used family of real-time qPCR system from Applied Biosystem. The QuantStudio family. One of the last version they have is the 12K Flex real-time PCR. It supports fast qPCR master mixes, which is optimized to deliver rapid qPCR results without compromising the sensitivity or reproducibility of an assay. It also has 5 interchangeable blocks, so you can read an open array plate, a Tachman array card, a 384 well or a 96 well, either 0.1 or 0.2 sample size. It also can support the multiplexing of up to 6 fluorescent targets, and also supports the LIMS system. So talking about the different PCR chemistry of the probe. One of the first ones is the molecular beacons. It's a stem and loop shaped hybridization dual label probe. The probe, once it is in this conformation, the fluorescence is quenched. When the probe undergoes a conformation changes, there will be an increase in fluorescence. Another type of probe is the Eclipse probe, the fluorescent reporter at the 3' end, and a quencher and a DNA minor groove binder at the 5' end. This is very similar to the ones we use currently in all our assays. When the probe is intact, it adopts a random coil conformation and brings the reporter and quencher together, so there's no fluorescence and quenching occurs. The probe hybridizes to the target with the help of the minor groove binding, and it thus becomes linearized, separating the probe and the quencher, and fluorescence take place. Another type of probe is the Scorpion PCR primers. Also, when intact, the reporter is quenched, no fluorescence there. It also has a PCR blocker downstream of the quencher to prevent any read-through during the extension of the opposite strand. The loop of the Scorpions, as we see in this picture, hybridizes to the internal target sequence, and the reporter again is separated from the quencher, and fluorescence occur. Those are there are other type of probes in the market currently, but the most two important ones we always work with are the widely used ones are the DNA binding fluorophore, the intercalating dye, cybergreen, and the hydrolysis Tachman probe. The fluorophore can be dual-stranded DNA binding dye, which is most commonly known as cybergreen, or covalently attached to the target-specific probe, as in Tachman assay. This diagram shows you how the cybergreen works. The dye fluoresces when it's only bound to double-stranded DNA. But it only displays very weak fluorescence if it's present in single-stranded. So anytime an amplification occurs, the dye intercalates in each of the double strand and results in an increased fluorescence as the reaction progresses and double-stranded DNA is constantly formed. The fluorescence is then calculated by the instrument. Another type of the hydrolysis probe, commonly known as Tachman, is hydrolysis dual label probe with, as you can see here, a reporter dye at the 5' end and a quencher at the 3'. Again, when the two dyes are in close proximity, the fluorescence from the reporter dye is inhibited or quenched by the quencher dye. As the Tachman DNA polymerase starts to synthesize a new DNA strand, it causes the degradation of the probe, and the probe is cleaved, separating the reporter and the fluorophore from each other, and the fluorophore is started to fluoresce at a distinctive wavelength, resulting in an increase in fluorescence emission. So the increase in fluorescence signals occurs each time the strand is duplicated, and it's proportional to the copy of the target produced. The instrument then quantifies the fluorescence signal at each amplification cycle and generates a real-time qPCR curve, where it's going to be plotting the cycle number versus the fluorescence, which is directly proportional to the amplification of the target. So there are 4 distinct phases in the amplification curve: lag, exponential, linear, and plateau. During the lag phase, the amplification occurs, but the signal is not strong enough to be a detectable level, and it cannot be detected above the background. The exponential phase is used for the quantification, in our case in qPCR, because it produces a fluorescent signal measurable over background and contains an abundance of reaction components that have not yet been used. So it permits a detectable doubling event at each cycle. So this is the phase where all the reading takes place in qPCR. When the fluorescent qPCR signal is detectable over the background fluorescence, a quantification cycle or a CQ value can be determined. So the CQ values can be used to evaluate relative target abundance of 1 or even 2 or more samples, something we call a multiplex, to evaluate the target quantities as well, in reference to an appropriate absolute standard curve, which we're going to see later in the talks how we design the standard curve. This slide shows you how to design a qPCR assay. This is an example of a multiplex RT-qPCR of 3 RNA viruses, the CTV, cirrhosis, and CLBV. What you can do is anytime you have to design an assay, you have to make sure to get the entire number of sequences in the gene bank, and as well as if you have the ability to generate your own in-house sequences, pile them up, and then design your 2 primers and the probe in the middle. In that case of the multiplex, each of the viruses under studies is incorporated with a different fluorophore. Here, FAN, VIC, and TET. There are different criteria to design a successful qPCR assay, which is you have to design a primer probe that are very specific and follow specific conditions. So after, as I mentioned before, after piling all the sequence in the gene bank or your own in-house sequence, you will use, we use usually an ABI, Primer Express software. You insert one of the pile-up sequence representative of all your sequences, and you design your forward and reverse and Tachman probe. The software will choose the optimum conditions by fulfilling these criteria. As you can see here, for example, the Tachman primers, they have to have a temperature between melting temperature between 58 and 60. They have to be 15 to 30 bases in length. No more, certain criteria have to be fulfilled. In case of multiplexing, we make sure that the amplicons of all the multiplexed viruses or pathogens that we want to multiplex have to be within a shorter amplicon size. The Tachman probe, for example, the melting temperature has to be 10 degrees higher than the primers to make sure that it attaches before the primers to the target sequence. Another criteria is the GC contents have to be, of course, higher so that the melting temperature is higher, and no repetitive, no Gs at the 5' end. So these are all the criteria to ensure a good design of your qPCR. The qPCR validation also, once you design and run the assay, you validate it by following these guidelines, such as the amplification efficiency, which is if the efficiency of the assay is 100%, this means that during the log phase of the reaction, the qPCR product of interest is doubling at each cycle. So we follow a specific formula for that of the efficiency. Another criteria is the sensitivity, which is the minimum number of copies in the sample that can be measured accurately within an assay. A pathogen load, in case of viruses, it will be the viral load, which is the measure of the number of pathogen particles present in an organism. In all these three criteria, the efficiency, sensitivity, and pathogen load, you do need a standard curve to determine the starting number of your copies. That will also be explained in full details later. Specificity also has to be measured, which is the capacity of an assay to detect your sequence rather than other sequence present in the samples. The evaluation using a variety of, in our case, citrus samples with known infection status of your pathogen under study and other pathogens in the samples. Other criteria are applicability, practicability, and transferability. By testing these assays at 2 different locations, you also test the robustness of the assay with a deviation of you test your assays in 2 annealing temperature plus or minus 2 degrees. Also different reaction volumes of plus or minus, and different qPCR instrument and kits in different labs. Another aspect is cross-reactivity, assessment using RNA of different non-inoculated citrus species and RNA from citrus inoculated with other non-target pathogens. Repeatability and reproducibility refers to the precision and robustness of the assay with the same samples in the same assay, in the same plate, or reproducibility refers to the variation in results between the runs in different locations as well. We also have to follow some MICE guidelines. Those are the MICE guidelines are the set of guidelines that describes the minimum information necessary for evaluating qPCR. It has been published for the first time back in 2009, and everybody should follow these if you want to publish a proper qPCR presentation paper. Recently, they have also created the digital MICE guidelines for quantitative digital PCR. In that case, they do require you to do a checklist to accompany the initial submission of a manuscript to the publisher, providing all relevant experimental conditions. As you can see here, for example, the primer sequence, probe sequence, the location and of any modifications. So you have to fulfill all these steps to be able to have a proper publication under the MICE guidelines. These are the steps and the variables for a successful qPCR detection. So any of these steps, you can have a proper PCR or you can really mess it up. So you have to be sure to follow the proper criteria, again, that will be mentioned in full details later on in the discussion, in the presentations. So you have to have a proper sampling. If you have a place that has if your virus or your pathogen is not uniformly distributed, you have to sample properly from all locations. Proper homogenization, the nucleic acid extraction, whether it's going to be robotic or column, the concentration, the integrity of your RNA is important, how you store your nucleic acid, whether DNA or RNA. The assay design, as we mentioned, the primer probe have to be the specificity of the primer probe and proper reference genes where you can use them. The efficiency of an RT steps, if you're doing, if you're starting with an RNA and doing and converting it to cDNA. The choice of fluorophores, what wavelengths they have, the qPCR platforms that you use. The kits, one of the main components of the kits is the type of the polymerase type in its own kits, the qPCR reaction component, and the cycling conditions. An example that shows you exactly how these will work is a proper sample processing, homogenization, RNA manipulation, extraction. For example, in that case, You can see that we did a mechanical disruption with a mortar and pestle. We placed the samples afterwards in a 1.5 tubes with lysis buffer, and then stored the samples at -80 in 1.5 ml tubes. Another, in the following reaction, we did a mechanical disruption with a grinder. We placed the proteinase K digested to reduce viscosity. We did a sample extraction with an automated method like the one we showed earlier. The sample was stored in -80 in 96 well plates. So as you can see here from the curves and the CQ values, those have been tremendously improved just by improving some of your steps of homogenization, extraction, and storage of your sample. This slide just shows you how these assays will streamline the rapid and high throughput detection in the CCPP. It shows you the steps starting from collecting your sample. As I mentioned before, we have to collect from different locations if our pathogen under investigation is not uniformly distributed. We put the samples in a homogenization machine instead of doing the mortar and pestle, as I mentioned before. You submit a subset of samples in these plates to the automated nucleic acid extraction. We use MagMax 96. Then you place your sample in any of these formats. You can place it in a 96 wells. If you have more samples, is the 384 wells, or in a Tachman card. And you put your samples and test your qPCR using the QuantStudio. Again, all these steps will be linked together by the LIMS database. So this is basically, again, the diagnostic workflow. You start by submitting your samples, and it shows you the steps that we take, basically emphasizing on the quality management. In that case, we use LIMS to make sure that we have the contact number. We produce a project ID first. And then, for example, you put the contact information, the project description, what tissue did you use, and the receiving and processing date. You later on give each of these samples an individual assigned sample ID with the name, the date, the sample species. You put the permanent homes. The permanent homes for each of the sample is assigned by linking the sample to one of these wells. While again, either use 96 or 384 well plate. You put the samples in a qPCR is then run, where the sample will also be linked to their corresponding diagnostic assays. Again, this is all linking the LIMS database or the quality management system that we currently do in CCPP. Thank you. And this is actually, the status of the citrus qPCR assays that we have currently in the CCPP. We have the singleplex and multiplex. We have a number of 30 pathogens that we work with. Some of them have been multiplexed together. They're design validated. We test them either by live samples or by oligos, and then we try to combine them in a multiplex, and that takes a long time for validation and testing. Thank you very much, and if I can answer any of those questions, I'll be happy to do so.
Is that possible we get the PDF file of all of the presentation? Would
For a cost, Saurabh.
Okay. No, it would be beneficial if we get it in advance. So when whatever you presenting, if you want to take the note, can we do it in our note?
See, those are type of comments that are very useful for us in our future training. We appreciate that very much, but we can still do that for the ones coming up next. We can send you guys what we have presented today, as well as the ones we're going to present tomorrow for taking notes. So if nobody has any questions, I can introduce Samantha Barnum. She is collaborating with us from UC Davis, and I let you talk, Samantha. Let me see if there's anyone who has any questions in the chat. Are positive controls available? Some of the positive controls are available, and we do test and validate our assays with the positive controls, live samples. If we don't have the positive controls, we do use oligos. We create oligos in silico and test them, which is the distance between the forward and reverse primers. As was mentioned here before, NCPN Center in Maryland can also provide a lot of positive controls. So we always seek their help if we don't have our positive control ourselves.
And if any of the centers needs any positive controls, just email us and we'll do our best to send you what we have. And as Fatima said, we are working. Roy is also relatively new in his position, and I don't know if you want to say something, Roy, but Roy is working to, he's tackling right now his greenhouse, organize his isolates and his plants, and he's going to gear up to bring in all sorts of newly discovered pathogens in citrus and so on.
We're actually, this is a good opportunity also to emphasize networking among our centers, among the citrus centers. Whoever wants any positive controls, they can shoot us an email. And as coordinators, we'll try to get the positive control. I mean, our first stop will definitely be Maryland, but if any other centers can help, we'll do so.
Why don't we do this? Because it's going to be very useful and guide Roy and myself, which I'm always asking Roy to help us out, and we're trying to help out Beltsville. Why don't we create a Google form, a Google Doc, so all of you participants, and spread the word, send the link out after we share, and you go in and tell us which controls you want. And that way we have a working list for Roy to apply for permits and for us to find contacts for Roy to get the isolates into the United States. Do you like this idea?
Actually, Giorgio, why don't we do it the other way? If everybody provide what they have in their collection, that list become more informative, and then
Both ways. Yeah, both ways.
okay, yeah, thank you.
Finish. Sorry, I got excited with the idea.
No, that was because if we know what USDA in Maryland they have in their center, then whatever we have, we can put it online, and then we can send it to Maryland, or we can ask them to send us.
Okay. So Fatima and Irene, an Excel spreadsheet with 2 tabs. One tab will have, and we can come up with the columns. One tab will have the name of the center, the name of the pathogen, the origin of the isolate, and if there is also maybe some gene bank information. And then the second tab will be controls requested, missing, that we need. So one is what is available, an inventory for the network to choose from. And the other one is what we need to bring in the network.
That's perfect. We'll set up a Google document with an Excel sheet like this. We have one for our CCPP that was created, but we'll take this suggestion and create this Google document and send it to all centers. That's a perfect idea, actually. This is exactly what networking should help us with our activities in each center.
Just from a technical point of view, we mentioned Roy, but also the other USDA centers. Sometimes they have trouble with the Google platform.
Oh yeah.
Sure. But that I just didn't want things to get lost in an email.
Sure. Yeah, that's why
what-
I was thinking if anybody has access, I believe it sometimes at USDA, they have access to Teams. And that I've seen that most institutions are able to participate in that.
Yeah, and
Something to bear in mind. Yeah.
yeah, and
This is Robert. I work
Yeah.
for the USDA, and usually if you share it to our USDA emails, it usually works out okay as far as a document type thing.
Okay.
Okay. Yeah,
Thank
and
you.
if there is 2, 3, 4 centers or people that they have trouble with Google, then we can get an email.
Okay.
But instead of having a
Of course. Yes. Mhm.
a slew of emails,
No. Mhm.
and then we transfer manually the information. Okay, that's important, and I always don't know where to start in my discussions with Roy what to tackle. Literature can guide you that much about new stuff and so on, but let's do an inventory of what we have and what we need, and we can move forward. You can capture this also on your report on this workshop that we are going to take this initiative.
Perfect. Then we'll follow up with we'll create this template and see what we have in CCPP and follow up with an email so that every center can fill up the list.
Yeah, and we can also link it. The Google is one thing, but Peter, our IT guy, can help to link to existing inventories and databases and see if it can become something more functional. Because it has always been the idea that whatever software and databases a program in the Citrus network develops, these platforms can be available to other centers if they want them. So this could be like an easy, low-hanging fruit to start sharing with each other.
Perfect. Samantha, would you like to start your presentations and give a brief introduction on yourself as well?
Fatima, sorry about that. Do you
Go
want to,
ahead.
you mentioned you want the name of the participant and the location for the report you need?
Yes, thank you for another reminder. In the break, we were thinking of if anyone would put their names as well as the institution in the chat box, that would be very helpful for us to for our reports as well to reach out to everyone. So if you don't mind, put your name and institution in the chat box. Thank you. Thanks, Zora.
You're welcome.
Go ahead, Samantha.
All right. So I've been tasked by Fatima and UC Riverside to give you guys 2 days of intense PCR training. I've been doing PCR for about 20 years, qPCR for about 20 years. I started with Fatima doing grape and citrus and fruit soon after I started in the real-time PCR core facility. I was kind of just thrown into qPCR. I really wish I had had a class, something like this, to give me a lot of background, someone that had more experience and was willing to share it. I was in a lab where you were kind of just on your own, and here were some basic SOPs, and good luck, and ask questions if you had to. Obviously, I was a new recent hire, just finished my master's degree. So you're kind of sometimes embarrassed to ask questions. And also the environment I was in, even though they said to ask questions, they weren't really willing to entertain them. And so as a boss, as myself, I think it's really important to let your staff know that questions are fine. Let's get to the answer. But that also, if you do good training in the beginning, you should have fewer and fewer questions later on through your experiments. So that being said, I'm going to start my slideshow here. So the first talk is going to be, what is qPCR? I'm just going to kind of start basic, kind of expand a little bit on what Fatima just spoke about, and then also get in some more detail. So the outline is qPCR, design limitations. I'm just going to touch briefly about sample extraction, 1-step, 2-step, singleplex, multiplex, because I figure especially your lab, knows how to extract samples and that type of thing. My background is in horse viruses and bacteria. And so I do know a lot about pathogen detection and isolation and quantification. And I'm kind of just bringing that right into your field of citrus. So quantitative polymerase chain reaction encompasses 2 different groups. One is called real-time PCR, and the other is cyber. Real-time PCR is frequently called TACMAN PCR. It's used very interchangeably. The TACMAN as a word is copyrighted by Thermo Fisher, so a much more broad or generic term would be real-time PCR. Probe-based would be your real-time PCR versus cyber. There is no probe there. One of the great things about qPCR is there's no post-PCR, production. You don't have to run it on a gel. You don't have to send it for sequencing. You just get the fluorescence and your CQ values. I will be talking about CQ values. CT is the old terminology. Fatima mentioned the Mikey guidelines. So that article said that CQ, cycle quantification, is the new terminology to use. Also, I meant to say, if you have any questions, please just feel free to ask them during the talk if you want, or at the end, whatever works for you. I'm totally fine being interrupted. So with qPCR, you're gonna get accuracy, precision, and your quantification, okay? How do you get those values? You're going to be doing a delta-delta CQ calculation, which is where you're going to get an up or down regulation of a gene. Not so much important in pathogen detection, but that's how most publications for genes, when you're looking to see if a treatment affects different genes, you're going to talk about your delta-delta CQ value. You can also do pathogen loads, right? You can do absolute quantification where you're going to say, I have 10 times 10 to the 10th copies per million cells, or whatever value you want to quantify against. You can also do SNP or genotyping where you're looking at a single base pair mutation or modification within a sequence. It's always kind of surprises me that you can detect with qPCR a single base pair change and get actual real data off of that. I just find that kind of cool. So what are some basic qPCR questions? You're going to start very general. What is your experimental goal, right? For you guys and me, it's finding a pathogen. Where is it located? What tissue type is it? Which plant is it in? For some people, it's gene up or down regulation. If I give this new drug, does it downregulate inflammatory genes? How many genes are you going to analyze? A lot of it comes down to money, right? How much your grant was for, what you put in there. Are you going to be analyzing your samples in singlet, duplicate, or triplicate? Do you have control samples? What's your time zero? Or do you have a negative treatment control group? What reference genes are you going to use? Do you need a reference gene validation study? Reference genes are critical in determining proper extraction and looking at inhibitors within your DNA or RNA. And so finding a good reference gene that's going to work for all different sample types or all different conditions is really important. What are your best sample types? Can you even collect them? I had this really cool researcher reach out to me where he wanted to look at biominerals from men's rest stops that were using a waterless urinal. Wow. You know, I'd never done any research like that before. And how does he go about getting into the rest stops along the highways to collect those samples? And would his pathogen even be there? You know, so sometimes you need to, some people really thinking outside the box. And I think that's where qPCR can really help answer some of those questions. So here's just kind of a broad overview of starting with your sample, preparing your DNA in one room, having a separate room for your clean room for stock solutions, a laboratory where you're going to mix everything together, your thermocyclers, your detection, how you're going to document it. Are you doing research and development? Making sure you have good quality control and a quality assurance guideline that kind of outlines the quality that you guys are trying to obtain within your labs. Like any technology out there, there are limitations, right? So some of the PCR inhibitors are big limitation in qPCR, particularly dirt, plant material, which you guys work with, hemoglobin, heparin, formalin, and how do you deal with those inhibitors? Does anybody have any ideas of what do you do if you think your DNA or RNA has inhibitors in there? What would you do to your sample or your total nucleic acid to see if you have inhibitors, and what would you do about it? So that's something I want you to think about. One of the main things there's two main things you can do about inhibitors is to dilute them out, okay? So normally, let's say you run 1 microliter of your DNA in your PCR reaction. Well, what happens if you do maybe a 1 to 20 dilution of your DNA and run 1 microliter of that? How does that change your reference gene? So really kind of cool study you can do within your lab if you really think you're having inhibitors within your DNA or RNA is run a dilution series of your DNA. Run it neat, 1 to 5, 1 to 20, 1 to 50, or something like that. And you should see the CQ value of your reference gene changing at each dilution. And then you can choose which dilution works best for that sample type. We also do that. We do a 2-step process in our lab. So you can also do that with cDNA. So when you dilute your cDNA, your final step, how much water are you adding? You can try adding an equal part of water, 40 microliters, 60 microliters. Again, run those with your reference gene and you can see your CQ values changing. Also, an inhibitor is ethanol. So when you're doing a column-based extraction, if you do not remove all the ethanol from that second wash and you elute some of that in your water, you're going to have inhibition in your qPCR. Just kind of general guidelines, you generally don't want more than 10% of your cDNA into your qPCR reaction, and you can put too much genomic DNA in there too.
Excuse me. Any sign for ethanol inhibitor in your sample? How do you know you have that one?
So how you would know is I would say if you get a lower CQ value of your reference gene than other samples that you extracted at one time would be a main clue that you have ethanol, that you potentially have ethanol in there. We do an extra drying step of our filter, and I think you could do the same thing with your magnetic beads to make sure that there is no carryover, but it would affect your reference gene and you'd have like a failing sample at that time. And it would just be one thing on your mental list of maybe there was a failed extraction. You're not going to know specifically that it's ethanol, but it's going to be in the back of your mind that maybe the extraction didn't work.
Thank you.
Let's see. You can also, I've never personally done this, but for some pathogens, you can use some dye and it's going to tell you if the pathogen is viable. Also, sometimes we will look at DNA viruses, and a clinician will want to know if that DNA pathogen is just there kind of in low abundance, or if it's actively replicating, and you can look at the DNA the RNA of that pathogen to let you know if there's replication happening. Some other limitations would be how you're storing your sample. Okay, are you putting it in RNA later? Do you freeze it right away in liquid nitrogen? I had another project where people were collecting nasal swabs from donkeys in Death Valley in the summer. So how are they going to keep that swab cool, right? So you need to think about your sample collection, storage, and how it's being transported, and how that's going to affect your downstream applications. Something else you always need to think about are the different types of extraction kits and enzymes being used. Testing different kits. I always recommend starting with a larger name brand company that's going to be around for a long time so that your kit's going to be there once you find it. Obviously, human error, right? Pipetting error, samples getting mislabeled, or someone can't follow a SOP. And being tired, right? So if you're not getting a good night's sleep, but you need strong attention to detail, you can be sorely lacking on that front in the lab. Regarding mislabeling, when I first started, we had handwritten tubes for all of our master mixes and primer probe mixes, and E stood for equine and C stood for canine. And some people were kind of sloppy writing an E and a C. So it was very hard to tell if a certain master mix was for the equines or for the canines. So mislabeling of samples or not being able to read someone's handwriting can definitely lead to errors down the line. So getting a nice labeling system is going to definitely save you time and energy in the future. How are you going to store your nucleic acid and where? Are you going to be storing your RNA at -80? Do you have -80 room? And how long are you going to store that? Are you going to store your nucleic acid for just a few months, or are you going to store it for a few years? The quality of your RNA. For qPCR, we're very lucky. We don't need the bestest quality to get good QC values in the end because our amplicons are so short. If you're doing next generation sequencing, you need very good RNA quality. So think about what your downstream applications are going to be, and that'll help define the quality that you need. Like any other kit, reverse transcription. Select a good kit that has a good DNA digest in it because you generally do not want to bring genomic DNA contamination into your following steps, particularly if your qPCR assay is not cDNA specific over an exon-exon junction. You will detect genomic DNA in your qPCR of your RNA if you have not digested it all the way. So assay development. I do a lot of assay development for the lab, and I do a lot of it with Fatima. So think about the chemistry you're going to use. Like she mentioned, you have to determine the qPCR efficiency of each assay, choosing an appropriate reference gene, determining if you want to do a singleplex or multiplex, what is the upside or downside for each of those. I'm going to go over that in this talk and in some of the following talks. Do you want to do a one-step process? Do you want to do a two-step process? How to analyze your data, how to choose appropriate baseline and threshold to meet criteria in your lab where everybody's using the same one. And hopefully that's possible for everybody's lab, because it will affect your CQ value slightly if you choose the wrong baseline and threshold. Particularly your threshold, if you choose it too high or too low, you can form false positives or false negatives, and I'll be going over that. It is basic here, like I mentioned, baseline and threshold, doing standard curves. Some publications or papers, you're going to need to put your inter assay variation, normalizing your samples against your gene of interest, against your reference gene. So I'm just going to go basic here over sample extractions, because there's so many different types out there, and it depends a lot on what your starting material is. So a very common chemical one is Trizol. You do that by hand, so it's not great for a high throughput lab like yourself and myself. Column filters, usually kind of by QIAGEN. Again, you can do those by hand or in automated systems. And magnetic beads. You can do those by hand or in automated systems. Some of it's going to depend on how high throughput your facility is. So Trizol, there's good and bad about it. It takes about an hour to do a Trizol extraction. You're going to get excellent RNA quality out, which you might really want for next generation sequencing. But like I mentioned earlier, you don't need that for qPCR. It's also kind of a cool system in that depending on which aqueous layer you take, you're going to be getting DNA, RNA, or protein. So I think that's kind of a cool system. Also, one of the negatives with Trizol is you're handling a lot of nasty chemicals. So it's not something I recommend. A lot of people use filtered systems. We use a lot of QIAGEN kits in our lab. There's obviously different brands out there, especially for RNA extraction. You're going to be adding beta-mercaptoethanol, which helps break up those disulfide bonds. Some kits use DTT, again, to break up the proteins that are in there. So you're going to lyse your sample, you centrifuge or pull a vacuum to get your sample onto the column. You're going to wash the column with 2 different wash solutions that have different amounts of ethanol and different amounts of salts. And then you're going to dry out all the ethanol, and then you're going to elute. We elute in DEPC water ourselves. We do not elute in TM or salt solution. So we have the instrument on the right here, the QIAcube HT, which extracts 96 samples at one time using a filter-based system. QIAGEN also has, I think it's a 12-well centrifugation system that's kind of a semi-automated. It takes an hour and a half for a full 96 samples to be extracted. One of the things I do like about this extraction type with columns is that you depend on what you want. If you want DNA, RNA, or total nucleic acid, you can choose the kit to give you that answer. in our lab, I need total nucleic acid. So I need DNA and RNA out of the same sample because I'm looking for different pathogens. I'm looking for RNA pathogens and I'm looking for DNA pathogens. And I don't want to take the time or the expense to extract the sample twice. So filters give me that great ability to get total nucleic acid out of a single extraction. Magnetic beads are very similar to a filter system, other than the DNA or RNA binds to the magnet. You can just drop the beads, which are minuscule, into a tube. You can mix them up and down, put your magnet in there, all your beads bind to it, take your magnet to the next wash solution, release the magnets, the beads go into the solution, mix them up, activate the magnet, and keep moving them through your different wash solutions. So it's a really cool process. It's a whole lot faster than filters. I used to have a biosprint in the lab, and I think it used to take us about 35 minutes to do a 96 well 96 sample extraction versus the hour and a half that it takes me now. Why we switched back to the filter base was that when I was using the magnetic beads, I was either getting DNA out or RNA out. I was having trouble getting total nucleic acid. And so that was a problem for our downstream applications. Here's just two pictures. So the picture on the right is the 96 well format. Thermo Fisher also has a 12 well format if you're just doing smaller number of samples. So that's how you're extracting your sample. I guess this kind of takes us a step backwards of how do you break up your sample. A lot of people use proteinase K. You can use physical grinding, either in a liquid format, like in the lysis solution, or you can also do it in liquid nitrogen. And some people use just straight up heat, particularly if you're looking at bacteria. Those guys break up really quickly with high heat. So that's just breaking up your sample. One step versus two step, okay? So it depends again on what you're looking at. So if you're looking at RNA and you want to do all your reactions in one tube, you're going to be doing a one-step cDNA synthesis, right? It's going to be in the same tube as your PCR. There's pros and cons to this step or to this technique. One is that you have to store the RNA versus storing cDNA. So if you're worried about freeze thawing or having space in your -80, that's something to consider. Also, when you do a one-step qPCR, there's not an opportunity to check that all your genomic DNA has been digested. So again, that's going to depend on your assay design. Oh, that's just my timer. Two-step. So that is when you're doing a cDNA synthesis first, and then you're adding cDNA into your qPCR. So you're having two separate tubes. This is what we do in our lab. That way we can store everything in just a normal -20. You can test that your genomic DNA was all digested. It's a really simple process. You just do your DNA digest, take a microliter of that out, run your qPCR with your reference gene, and it should be negative, right? There should be no genomic material in there. We found when we were doing plant samples in the lab that they have a lot of genomic DNA in there, and different enzymes, different DNases were better or worse at digesting that DNA. So please take that into consideration about your different sample types and their DNA availability in there. This is just kind of a synopsis, pros and cons. One step, you're going to save a little bit on tips, right? Because you don't need a separate plate or a separate tube to do your cDNA synthesis in. Also gives you less chance of having contamination because it's all in one tube. It is better for high throughput labs because it's one less step to do. I don't know this for sure. I just found this slide. It says it can be less sensitive. I don't have any proof on that. 2 steps, you can use random primers. You have your stock cDNA that you can reuse multiple times, easier to freeze thaw. PCR inhibitors can affect your cDNA synthesis. And then this also says sometimes cyber has high background, but I don't know about that. Singleplex versus multiplex. I only do singleplex in our lab, so each well has one target in it and one gene of interest in it. So on the 384 well plate, that's how many reactions I can have. I find them easier to validate. It's just one standard curve. It passes or it doesn't pass, and you go on from there. I also think it's a little easier to analyze because you're just looking at one fluorescence. You're not worried about any interactions between your different fluorescents. Multiplexing, I've done the 3-way multiplexing for your guys' lab. You can go up to even more than that. I think you can go up to 5 or 6. I've never done that. You're going to have to be seriously considering the interactions between all of your fluorescents and your quenchers. So you're definitely going to want to choose detectors that are spaced far out in the spectrum so they have less interaction upon excitation. It's a little bit more challenging to validate and to set up the assay design. And I think they're more challenging to analyze because now you're looking at 3 curves and how do you set parameters where all of that is happy at one point. And I'm going to have a whole talk on that multiplexing. So I'm going to be showing you specific examples of nice looking curves and not so nice looking curves and what do we do about it. So that's my last slide for this talk. Is there any other question? Yeah, Georges?
Yeah, Samantha, when you talk about the curves, please remember when you show us different curves, I knew it at some point, I figured it out, but I was always not confused, but there is the normalized sigmoid curve that all of us are used to look at, but then there is the curves behind that nice sigmoid curve that it's really jumpy and pointy, and you know what I'm talking about?
Yes, yeah.
There are the reaction, okay, and then whatever software, if you can, when we talk about curves, if you can walk us through that, how one represents, I think, the actual dynamics of the reaction, and then everything gets normalized to become sigmoid. If you can explain that to us when we go to the curves, I would find it refreshing for me.
Yes, absolutely. Yep, I have that one of the, I think this afternoon we talk about that.
Okay.
Sorry? Yep.
Okay. About the 1 step versus 2 step in diagnostic, pathogen diagnostic, do we ever do 2 step from the RNA target and RNA pathogen?
The twister like on your PCR machine?
No, what would be the reason if you do in the detection laboratory and you are targeting RNA pathogen and your sample is RNA, have we ever run 2-step RT-qPCR?
I don't know if you guys do. I mean, we do in our lab. We always run 2-step on those guys.
Why?
Why? Because I like
what would you accomplish by doing a 2-step? Only you mentioned about the storage, so you get the cDNA, but you make your workflow twice more. Cross-contamination comes and play. So what would be the benefit?
The benefit for me is so now that I've made cDNA and I have that storage, I can store that at -20 for months and months. And if I have to go back to retest that initial sample, my cDNA is very stable. And so I don't have to worry about any degradation in the freeze thaw of that cDNA to retest it again in the future. Also, I think personally the enzymes that are making that the reverse transcription, I think the enzymes are a lot better in a separate kit versus already mixed in there with your PCR reaction. And then also, like I mentioned, checking for genomic DNA contamination. Because if you don't test for it, how do you know you have it there or you don't have it there?
Okay, so that's what I was emphasizing. We are doing the pathogen
Right.
diagnostic. It shouldn't be there. It shouldn't be any genomic from the host or your matrix there. So digesting the matrix background DNA wouldn't be an issue for diagnostic. But when you use these two steps, would you do a randomized primer or you use a specific primer for cDNA?
No, I would recommend using random primers because that way you can go back and do retrospective studies and
So
go
you-
back to your freezer.
Yeah,
Mhm.
sure. So you make the library. So then whatever probably is there, you are making as a DNA copy.
Right.
So one more question about the QIAGEN platform you show. You show two systems for the less than 96 format,
Mhm.
24 or 12. Which one you recommend for the lab?
Always is going to depend on how many samples you're normally processing in a day and where you see yourself going in, a year or two. I would always recommend the larger system because even though it's a 96 well format, you can set up your plate so you only extract what you have for that day. So if you only have 24 samples on a Monday because it's your slow day, you can block off the rest of that column and reuse it in the future. So
Okay.
I kind of tend to recommend going with the larger one if it can be afforded.
Okay. So right now you can cover because before they said they cannot. Either you use it and you lose the whole things. So that's so with this new one you can actually cover 2 or 3 rows. So only use the top one and then come back and use the rest, right?
Absolutely. Yep.
And when you mention about total nucleic acid, all of the QIAGEN are very specific, either RNA and DNA. Which kit you use with that with the column with the system that you get both DNA in the plant?
Yeah, the QIAGEN's DNA Easy kit, the blood and tissue DNA Easy kit from QIAGEN, actually a lot of RNA comes out in that kit, and that's the kit we use.
Okay, so that one has a DNA's, or I'm sorry, RNA's treatment at the beginning. If you take that one out, I'm doing it that way. So presumably you get RNA and DNA in both coming out of the column, correct?
Correct. Yes.
Okay, thank you.
Yep. Any other questions?
With regard to choosing two-step versus one-step, I prefer two-step because we always are interested in testing for multiple pathogens. If you consider cost of one cDNA reaction versus multiple PCR versus multiple two-step reactions, cheaper to do two-step reaction. And like you said, many other advantages. I agree with those.
Yeah, thank you. I appreciate your comment. Yeah. I think some of it also is like, what you're familiar with doing in the lab or what your PI or your director is asking for you to do. our lab has always done a two-step process. I think so. We kind of just always start there, but we do it for different.
The other thing is, if you have a RNA extraction, make a cDNA, then DNA pathogens do make RNA. So you can detect, if you design your primers and probes correctly, you can detect both RNA and DNA pathogens using RNA extraction itself.
Yeah,
Even-
if the DNA comes out during that extraction, some kits are better at that than others.
No, what I was saying was, no, if you just use RNA extraction, assuming that your pathogen is alive when you collect the sample, then your RNA, if you target towards genes that are expressed like pathogen genes are expressed like 16S or something else that is expressed at higher levels, then you can detect both RNA and DNA pathogens. That's what.
Sounds good. Any other comments?
Actually, Samantha, we did an experiment and we wrote a paper on comparing between one step and two steps. We used like a few kits that are in the market versus for one step and two steps, and we were testing at the time grape viruses, and the results were very comparable, and sometimes the RNA kits or one-step kits give us slightly better CQ values. That's why we were going through with all our testing using the one-step qPCR. So I guess any application in any lab needs a little bit of validation before you decide as well. So this is one of the parameters. Yep. We wrote the paper with you guys.
Yeah, no, I do remember that after you brought that up. Yeah.
Yeah, and we actually also did some experiments on treating with different DNAs, treating the RNA and DNA with different DNases before we start and things like that. So those are valuable information. Thank you, Samantha. Go ahead.
All right, let's go on to the next talk here.
Sorry, one more comment. If you take your RT out of your qPCR, RT-qPCR, you accommodate whatever you just mentioned. So having it, if you don't have it RT and then you get the result, then you get that target has a DNA in it. So then still you can do one step RT-qPCR, having RT as a one row or not having RT, then you compare your result, the CQ value. I hope it makes sense.
Yeah, so you're just saying that wait, can you repeat that?
Yes, if you have RT-qPCR reaction, you want to see there is a background target as a DNA entity in your sample. By adding or eliminating RT from your master mix would accommodate whatever we need to know if the target has a DNA background or DNA from the host or is only RNA target. And with the designing primer targeting intron or exon, then you can accommodate the same thing.
You could, yes. Yes.
Oh, thank you.
All righty. Next topic here. How to design primers and probes for quantitative PCR. This was one of my first jobs in the lab when I first got hired. It was not for you guys, but I had to design 50 different genes for tomato plant for assays, and I was given the software of Primer Express. I was given the sequences for all the different genes, and I was told to design it. I was given zero guidance or guidelines that are used for assay design. And so as I'm going to be talking a lot about different guidelines, I want you to keep in mind in the back of your head that that's what they are, is they're guidelines, right? So they're not rules. They can be broken. I wouldn't recommend it. You're probably going to get lesser efficiency, or your assay may not work. But I didn't know any of these when I first started. Like I said, I was poorly trained. And so I was just making sure the TMs were good for the primers. If it had 6 consecutive A's, oh well, I didn't know any better. I designed the assay. So out of those 50 tomato genes, my pass-fail rate, if you will, was about 90%. So 10% of the assays did not work. And I'm assuming at this point in time, in hindsight, is because I wasn't following any of these guidelines. My pass-fail rate now, I'd say I'm about 98% pass the first time. I do sometimes have to redesign an assay, and then they usually pass the second time. So these rules or guidelines, keep that in the back of your mind. You're going to just have to jump in if your job is assay design. Just go for it, play around with things. It'll generally work out in the end. It's not as scary as you first think. What is a little bit more scary is when you have to multiplex, because there's so many values that need to be similar between your different assays. Your amplicon length, your GC content is all critical for multiplexing assays, and I'll go over that when I do my multiplexing talk. So I'm not going to, this is not just tailored towards plants. This talk was initially for another PCR class, but it's going to go over how to design your own, aligning sequences, validating, things to think about for troubleshooting, and resources that are out there for you to use. So when I first get tasked with designing a new assay for a project, I generally will look, for published assays already, right? Why invent the wheel? Sometimes I'll look for just conventional PCR assays. Those generally don't help you too much because the amplicon is too long. Also, one other thing about published qPCR assays is alwa' verify everything that's published. You will not believe how many times things are not in a 5' 3' orientation, or the reverse is not the reverse complement, or the probe is on the antisense strand, so I can't find it when I blast it. The TMs are not what they say they are in the publication. So always verify any assay that's published. Don't just go order it and think, oh, it passed assay validation in this paper, I can just order it and use it right away. Absolutely not. You potentially will get in trouble with your results in the end. Also, I like to look at Life Technologies. They have a lot of, for different species, generally obviously not plant species, but they have a lot of pre-designed assays out there for you. PubMed and NCPN, you're going to have to be very familiar with that platform for blasting things and looking for your sequences. And then also Ensemble is another great website that is I use frequently for exon-exon junctions. They have an extensive list of all the genes in there, and it's really easy to distinguish where those junctions are if it's important that you make a cDNA specific assay. That's something you have to think about in the beginning. What are you using your assay for? Okay, are you using it for gene expression, SNP determination? Are you doing pathogen detection, just a straight up positive or negative? Do you want it species specific, or do you want it a generic, multiple species to be picked up? Does it matter if it's genomic DNA specific or if it's cDNA specific? So these are all things you need to think about before you start this process of designing. So this reiterates a little bit of what Fatima talked about. I'll have to say that the TM, the melting temperature of the primer and the probe, I am 100% firm on. I never go higher or lower than the 58 to 60 degrees for the primer, and for the probe from 68 to 70 degrees. That can't ever be changed because of your cycling conditions for your PCR reaction. I do try to keep the GC content kind of in mind. The Primer Express software gives you a green number if it's within the acceptable range and a red number if it doesn't. And the same thing with the length. Sometimes the length can go a little bit longer if you have a really AT-rich sequence. The Primer Express software is a one-time site license, so it's per computer. But then whenever you update your computer, or get a new one, you can still transfer that same license. So it's a one-time investment. You only really need it on one computer within your lab. You don't have to have it on for every technician. So I would totally recommend getting that software or another one, but just choose one software and use that for your lab. Generally, like I said, no more than 4 base pairs and the same base pair in a row. For probes, you generally don't like a G or a C at the 5' end. The shorter the amplicon, the better you're doing. You can check for secondary structures of your primers and your probe within Primer Express. And you'll always will have to blast your amplicon. And I use that at NCBI, and that's the best way to look for any mismatches. Published conventional assays are not too helpful. Like I said, the amplicons are too long. Sometimes I will use the forward or reverse primer as one of my primers for the qPCR reaction. One thing it is useful for is giving you maybe an example of which gene you should use, because sometimes that's even, you're not sure which gene to start with. So sometimes that gives you a hint on where you should start. Life Technologies has quite a few gene expression assays that are commercially available. If you do order from them, they're not validated. They only have been put through pretty much Primer Express, where they just know everything should work together. You do get the probe location, and if there's an exon boundary, you do not get your primer sequence or location. They're pretty reasonable. And it's a great starting point if you want to know which exon you should look at to design your assay. So if you go to their website and you say you want to look for tumor necrosis factor in a mouse, it's going to list all these different assays that they have for it. You get your sequence number, you get your amplicon length, and you just, also you get your aliases or what that gene is also known for. Here you can see where the exon boundary that they give you right here. Oops, sorry. They can also tell the amplicon length. It just gives you a lot of information to start with. When you need to design your own, you need your sequence, right? Generally, Fatima gives me that sequence or the pile-up, so I know she's already done a lot of that groundwork for me, which is awesome, but sometimes you'll need to do that yourself. Also, you can just plug that sequence right into Primer Express, press that little green arrow, it'll design an assay for you. Same kind of conditions for primers. and just you have to blast that amplicon in the end. When you go to NCBI, you can plug in the gene name, or if you know the gene bank number, you can put that in, and you can get the sequence like here of that gene, and you can just copy and paste that right into Primer Express. So when you open up Primer Express, the first thing it does is ask you what kind of probe you want to use. Do you want to use a minor group binding probe from Thermo Fisher? Yes, is your answer. Do you want to use a regular quantification probe that does not use that minor binding group technology, which means you're going to have a slightly longer probe? Or do you want to do allelic discrimination like a SNP? So generally, you're always going to choose the minor group binding protein choice here. You just paste your sequence in there, and then you're going to press this green arrow, and it's going to find primers in a probe based on the logarithm that Primer Express has put together for you. But we've talked about these rules or guidelines regarding A's and 4 A's together. And if you look down lower right here, you can see its own software has chosen a probe that has 5 C's together and 4 A's together. So sometimes the software even breaks the own rules. That being said, I would not choose this assay because of that. There's this little tool right here, Primer Pro button, and when you click that, it lists about 250 assays or combinations that the software has designed for you. So when you click that button, it lists all these different assays that it found. Also over here, you can look at the hairpin or self-dimer or cross dimers between your forward, your reverse, or your probe. So if that's important for you to look at, you can, do that individually there. Sometimes you will press that green arrow and it says, you press that green arrow up here and it says, no acceptable primer probes found. And that's when you kind of mutter under your voice, because now you have to do it all by hand. Okay, so you have to find your own forward, your own probe. You kind of really going to just be flying by the seat of your pants. I will give you a little hint or useful tip that about 10 G's or C's will give you a TM of 60 degrees. So I kind of just run my eye over the sequence, looking for a little bit higher GC content area. And then I just start, count 10 G's and C's, copy that sequence, and then paste it over here into your forward primer. And then it gives you the TM and GC content. You can see these are green, which is a good thing, which means it meets the guidelines set within the software. And so that's a good first primer. So you're just gonna, choose each of your locations, copy and paste it here, make sure these are all green. And then the final thing you're gonna do is blast that amplicon. Also, what's really important that when you're checking primer sequence TMs is that you use one software for the lab, because different softwares will use different logarithms to calculate that TM. So even if you don't want to use Primer Express, just choose one software or one website for your lab, and that everyone is, knows that they have to put their primers or probes through that software so that you all have similar TM calculations. So here's kind of an example of, here's my primer, got it from the paper, but my TM is low. But I really like this assay, I like the author, let's say, for example. So I just want to tweak this assay a little bit to make it work for my cycling conditions on my PCR machine. So I'm just going to start by adding, maybe I'll add this GC here at the end to this end, and then just see how that affects the temperature. And that's all you need to do to increase or decrease the melting temperature of a primer to get it to work. So you can see I added the GC here, and now my TM is good. So now I can use this primer. You still, again, want to blast it to make sure everything's working well. Their Applied Biosystem has a TM calculator tool. If you wanted to use that, they have their website that you can go there. And again, you just kind of plug in your sequences, and it gives you all the information here about TM and length. It's just another resource out there. You can do whatever you want.
But most
Yes.
of these software require what kit you use, because the concentration of the salt can drastically change or increase your melting temperature. How would you consider that one when you are using the software?
No, I've never had one of the softwares address that using different salts in there.
Okay,
I
just
think
go
that's
back.
also,
Just go one slide back. In your slide here, you say salt concentration assuming 43 nanomolar. If this is concentration of the primer, but there are, even you go any of the kit you go, they ask you what kit you use, because depending of the RT and DNA polymerase concentration of the salt, magnesium chloride or manganese, then the melting temperature shift. The annealing temperature.
right. That's why we only elute in water. We don't elute in any other, in a salt concentration.
No, your master mix has that concentration. If your salt concentration in master mix vary, then the temperature for your annealing is going to change. So how would you consider when you are designing it?
I don't take that into consideration at all.
Okay, thank you. So, okay, perfect. Thank you.
I've mentioned blasting a couple times. So BLAST is an acronym of Basic Logical Assessment Search Tool. So when you're BLAST in NCBI, you're searching for your sequence versus all the other sequences within their database. I would always recommend blasting the whole amplicon, not just your primers or your probes all individually, because they're going to be working together as a unit. You're not going to be just having your forward primer bind. Also, you want to see if there's other similar amplicons out there. If there's mutations, are they between your probes or your primers so it doesn't affect the binding of your primers, which is ideal if your assay, particularly some of your plant viruses, that they mutate a lot. So if there is mutations, you try to put those between the forward and the probe, or between the probe and the reverse primer, because that won't affect the ability of your primers and your probe to bind during the PCR cycles. So you just copy and paste your sequence into this box. You can say to exclude or include only specific pathogens. When I usually try to include everything because I never know what else is going to be in my sample. So I'm not going to just say only look for the CTV virus. I want to look for all viruses because maybe this assay is going to cross-react with a different virus. And you want to know that now. You want to know that before you order this, that this is going to react with 2 viruses and you're going to have to redesign it. Down here, I didn't catch this in the screenshot, but you can scroll down a little bit longer. And the normal default is 100 sequences that the BLAST gives you of similar genes, but you can't increase that to 250 or to have 500 hits for the most similar sequences out there. I do use this exclusion feature when I want to see particularly that if there are other viruses that it will cross-react. So I kind of generally do 2 blasts. I do 1 blast where I detect anything out there that the sequences have been put into NCBI. I get those results. If they look good, I do a second blast where I exclude my sequence to see if there's other similar ones. Because sometimes if that pathogen has been highly sequenced and there's a lot in here, you might get 500 hits just for that CTV virus. But what if the 501 is a different assay, or sorry, a different pathogen? So sometimes I'll do 2 different blasts to look for everybody and then to be specific just for my pathogen. So you get this cool little graph here that shows, you have 100% homology here, 100% coverage. These all are matching 100%. You can see these ones are only matching at 93%. So generally, maybe that whole sequence of that gene might not be known. You can click on any of these accession numbers and it'll take you to the alignment. And this is kind of what that looks like. So I've kind of just put on here, here's my forward primer. This is a really short probe and my reverse primer. This was one of those ones that only had a 93% hit because the first 7 base pairs of this gene are unknown, right? They didn't, when they were sequencing it, they just didn't move their forward primer too far out here to the left. And then here is not a mouse species, and you can see we have a few mismatches on our forward and our probe. So then you have to weigh the chances. Do you think that this forward primer is still going to bind with 3 mismatches here? If this species, for you guys, if this species of virus was in your sample. So you have to weigh that thought. Taking a big step back here, if it's really important for you to have an exon or a cDNA specific assay, I really like to use this website, Ensemble, that helps me look for those junctions. You can just plug in your NCBI accession number here. It tells you right here that this sequence matches it. You just click on it. I know this is a little confusing. I'm going kind of quick because for your plant stuff, I don't think they have plants in here, but I just wanted to give you an example of how you kind of go through this process. So here it shows each exon, and here's your ATG start site. So if you wanted an assay that was specific for cDNA, you could start maybe putting your forward primer or your probe here and then continuing it on here. So that makes sure that this assay will only detect cDNA. If you have any DNA contamination of this pathogen, that it will not cross-react with it. So that's how you look for your exons, and that's also how you make it specific to it. It doesn't matter if you put your primer or probe over that junction, whatever makes you happy that day. Sequence alignments. Like I said earlier, Fatima gives me sequence alignments with, I don't know, sometimes it's just 5 sequences, sometimes it's 50, and we have to paste them on the wall so we can see what the heck is going on with these different viruses. So they're really nice if you want to make sure your assay is specific for that pathogen. Or maybe, for me, when I first started doing assay designs, I was doing it for the horse. The horse hadn't really been sequenced yet. And so I was making sequence alignments for like the dog, cat, mouse, and rat, and kind of choosing areas where there was high homology and hoping the horse would be very similar. So you can use the alignments for 2 different ways: to exclude sequences or to make sure you include them. Okay, so it just depends on where you want to go with your assay. Fatima and I used to use Vector NTI all the time. It was a wonderful software. Unfortunately, it's no longer supported, but it's a great tool to use to align all your sequences. You kind of just copy and paste them into the software. You can choose that I want to align these 2 mouse sequences of tumor necrosis factor. And then you get this really cool breakdown. All the yellow is homologous. Here's your little mutations that are different between your mouse and your rat. So if you wanted an assay that's going to work in your mouse and the rat, you didn't want to set, you wanted one assay that's going to detect both these viruses. You don't care which virus it is. You just want to know if that virus is there. You're going to put your one of your primers and your probes here. Now, if it matters to you which virus this is, you might want to put your probe where you have these 3 different mutations to distinguish between your 2 different viruses. So the alignments are great to do the 2 different functions. This is just reiterating that. You generally, if you want a species-specific assay, you want as many mutations between your primers and your probes as possible. 2 would be a minimum on your probe, but the more the better. Generic PCR probes, so these would be ones where you could get from other websites other than Thermo Fisher. They're generally a little bit longer, and you can still use different detectors like FAM. Your quencher is generally Tamra or dark hole quencher. I particularly really like the TACMAN probes from Thermo Fisher because they're shorter, and since they use that minor group binding protein, it enables them to make that shorter probe. And it's really nice when you have an AT or GC rich sequence that you can squeeze in this probe a little bit easier, you know, than a longer probe like this. The price is very comparable between the 2, so it's not really something I worry about. So now you've gone into all this work of designing the assays. So now you need to validate them. Right, now you want to make sure your primers and your probe and your nucleic acid are all working together. So all your primers and your probes should be all reconstituted to 100 picomoles per microliter. If you can also buy some probes and primers that are already reconstituted, and both of them are in a tube when you buy it. It just depends what you want to do. You're going to need a positive control. So you can have a clinical sample, experimental, a long oligo or a plasmid. You have lots of choices for controls. When I start with the new validation, I run one single well of my positive control and one single well of my negative. A, I want to make sure the negative is negative, and then B, it's nice to run a single well of your positive to know how strong it is. Depending on the CQ value of that control will determine what concentration or fold differences you will do in your standard curve. I'm going to go over that in just a minute. You do a 6-point standard curve in triplicate is always your starting PCR reactions. What you're going to get from this standard curve is your slope, your R value, how well you pipetted, the efficiency of the assay, and the sensitivity of it. For our lab, the efficiency of the assay must be greater than 90%. If it's under 90%, the assay has to be redesigned. So each lab should set its own cutoff of what is considered passing or not passing.
Sorry, for each assay you develop, do you usually develop one set of probe or several and run it at the same time and choose the best one, or?
Nope, just one.
Okay. And if it fails, then you go and design another one.
If it fails, I design new primers are usually my first choice because they're cheaper and they come quicker and the probe is expensive. So if it fails, I usually will design primers, you know, more 5' of the forward and more 3' of the reverse.
Thank you.
Mm-hmm.
May I ask one more question?
Yes.
You talked about efficiency should be more than 90%. What is the higher limit?
Good question. I've gotten everything from like 100 to 102 to 105. I don't always know what those mean. I generally, when you get those really high ones, your sensitivity has gone down. So I still will redesign those. But if it's 100, from 90 to 100, I take that as passing. If it's over 100, I will either rerun it and hope it goes lower, or I'll redesign the primers.
Samantha, I think from our, Sora, maybe you remember better, from our training with the group from Europe, AATT is their name. I think they're involved with the Mikey. I think they said anything above 105, something is wrong.
Okay.
Yeah,
You
it is
shouldn't
90
go
to
more than
90 to 105. Yeah.
90 to 105. Okay. Thank you. I'm
Yeah.
corrected. It just doesn't happen too often, so I'm not always up on that type of thing. But thank you. So here are two graphs. This one here on the left is a 2-fold standard curve. And this one over here is a 10-fold standard curve. I always plot the lower CQ values on the bottom so that you can get a nice curve here and determine your y-intercept. You're always going to want your y equation and your r squared displayed so that you can plug them into this formula down here. One of the things that is not good about this example is that neither one of these are going negative, okay, going to 40. And it's really important to make your dilution series past where it should be negative, because I have had 1 or 2 assays where I'm assuming there's primer dimers or something like that, where it never goes negative. It varies between 38, 37, 39 and a half, 38, 37 type of thing. So our lab standard or SOP is that you always have to have this last point go negative to prove that the assay actually does that. When you take this slope value, this 3.4, and you plug it right into here, you get this efficiency, okay? And you change this 10 here to a 2, depending on the dilution factor that you use. So you could do a dilution factor of 2.5 or 5.9. It doesn't matter to me, but you just need to make sure you plug it into your equation and that you make your dilution in your tubes accurate to reflect a different type of dilution series. I would always start with the 10-fold if your positive control is strong enough, because it's a little more forgiving on any pipetting area. The more a 2-fold standard curve, you're going to show any pipetting error much higher because you're wanting a CQ value or difference of your slope right around 1. Okay, so if you get 0.9, you might not be passing your standard curve. Also, if you do have an outlier, it is okay to delete 1 point out of your replicate. For our lab, our minimum is 3 points on the standard curve with 2 points of your replicate. That's the very minimum that we can delete to make a standard curve pass. I try really hard not to do that because I want to see all the variation and the dynamic range of your data. Yes, Georges.
Samantha, any tips on how to make sure you are pipetting properly?
Yeah.
When you make it,
so
yeah.
So it's one of these things. When I learned to make a standard curve, I was told to use the same pipette tip to move from tube to tube to tube. My colleague Evelyn in the lab was told the complete opposite, to use a separate tip every time you move your dilution series over. So I kind of would recommend, using the same tip. So I set up my 6 tubes. I put 90 microliters of water in each one. I put 10 microliters of my positive control into one. I then vortex it. Kind of, you can either just flick down the DNA to the bottom of the tube or you can centrifuge it. I use that same pipette tip, mix it once or twice, and then take that 10 microliters to the next tube. And I just repeat that process. I like to do the 10 plus 90. Again, it gives you a little room for pipetting error. You know, since you only really need 30 microliters, you could do, 5 and 45. But the larger volume gives you a little bit more room for pipetting issues. You can also, some people will, people swear by different things in different labs. Some people are going to want to add a little BSA, a little protein in there. Some people say you need to heat it up, heat it up to like 85 degrees and heat up your water and then do all your mixes. I kind of try to only employ those little tricks when things are not working. I just try to keep it really simple and try to include as many data points when I first start. Soraya? Do you have a question?
Yes. So you mentioned how many, I'm sorry, I lost my school of thought. So usually where do you get those data point, a scattered data point that you need to delete? Is it toward to the 35 to 39 and upward?
Like how do I choose which ones to delete if I'm not getting a passing standard curve?
Correct.
Good question. I usually start, so if it's this example here on the right, I would probably try deleting these two bottom points, because how often am I going to get an actual biological sample that has a CQ value of 20? Not too often, right? You're normally going to, especially from pathogens, you're usually on the low end. So I try to delete. I usually will start at the stronger end, maybe delete a point, see how that affects my data. And then often I will potentially delete a data point at the low end, because I know that's where I'm going to have my greatest pipetting error. And then I'll delete that. So sometimes I just go back and forth, and I'm just constantly looking at the slope and the efficiency.
Thank you.
Mm-hmm. Troubleshooting. Okay, so we're getting down here to the end. If your sequence is AT or GC rich, the software is never going to find an assay for you. You're going to have to design it yourself. I would honestly look potentially at a different gene, if possible, to get away from that, those sequences. We've kind of touched the bases on this. What if your assay does not pass efficiency? You know, redesigning your primers. I've hardly had one ever fail the second time. I would never, ever, ever change my PCR cycling conditions. And I would never, ever change the concentration of my primers and my probes. You will see that frequently out there in publications or troubleshooting guides to try different concentrations of your primers and probes. And I cringe every single time, because if you do that, you have to keep track of it. So you have to know that when you run CTV, it's going to run at different cycling conditions than when you're running HLV. Okay. So that means now you cannot run those two pathogens on the same PCR plate. And you have to keep track of that. Same thing with primers. One assay, let's say you use the primers at half strength, and this assay over here, you use it at double strength. And then you have another one that you use at the normal 100 picomoles per microliter. You're going to have to document that and keep that straight for each assay. As a high throughput lab, that's a nightmare. I have about 3,000 PCR assays in my freezer as we speak. If they're not all run at the same PCR conditions and same primer probe conditions, I think I would shoot myself. That would just be way too complicated to keep that all straight. I rather redesign an assay than change my PCR conditions or change my primer probe concentrations. Now that's how our lab handles this issue. Everyone's going to be different. So we do not do any optimization of concentrations, okay? It either passes with our conditions or it doesn't. I'm not going to play around with it. I'd rather redesign it. What if the sequence is not known? Like I said, you can look for regions of similar homology between other species. Some other troubleshooting issues, like I said, using the same software to calculate your TM. I've talked to labs where every graduate student uses a different software. And then they come to me because they're having problems with the efficiency when different people run things. So choose one and use it. Sometimes you're not going to know where your exon-exon junction is. You know, do you need to worry about DNA contamination or your DNA digest? These are just choices you're going to have to make. A big one here, right? What if your what if your negative control is positive? It is generally due to some form of lab contamination. I would throw away all my waters and kind of start fresh. I have a whole nother separate talk where we talk specifically about this, but it's something, you know, you always need to run that negative control water to make sure it's negative. And then I'm gonna go specifically on how to swab your lab and look for contamination in one of the future talks. What if your blast fails? What if you blast your amplicon and it's picking up multiple viruses, not just the one you want. Okay, you just choose a different location or choose a different gene. Make an alignment so that you know where to design your assay where there's lots of mutations where it's not going to pick up. What if you can't get a strong positive control? Nowadays, it's very easy to buy a long oligo. When I first started 20 years ago, that wasn't really too much of a thing. You either had to use a biological control or a plasmid, which cost money. So sometimes we would do conventional PCR or pre-amplification of a weak control to make it stronger. I mentioned earlier, don't trust publications. You know, double check the blast. Quite frequently, you know, the reverse primer is not the reverse complement. The probe is the reverse complement. So when you are copying and pasting from a publication onto your sequence and it can't find it, you're first going to be thinking, what the heck? Why this is in a publication? So this primer has to be here. Try taking the reverse complement or the reverse of it or something like that to find the sequence, because you know it's there. You just got to manipulate it because they published it not in the right format. Final remarks, and then we're definitely, Fatima has a little bit of a talk and then we're going to do lunch, is that in the beginning, assay design kind of either goes smoothly or it goes very it's very frustrating. It kind of just chooses one route or the other. I don't know what it is. So I would really recommend setting time aside without interruptions. So that you can concentrate this. Don't be answering emails, don't be answering texts, you know, set some time alone where you can concentrate on this. Use your resources, you know, literature out there, websites, all that stuff is going to help you find the right primers and probes for you. And then lastly, you will get better and faster with time. It's like anything else out there. It's practice, practice, practice. I can design an assay now like in 10 to 15 minutes versus it used to take me half an hour. So it's just, you'll get better with time. Just jump in, try it, run your standard curve, and then change things from there. So that's my talk on primer probe design. Looks like Saurabh has a question or comment. No? Okay. Anybody else have any questions or comments? Yes.
Sort of again here. So you mentioned we cannot blast if your probe sequence is if the target's negative sense RNA, your probe is on antisense. We cannot blast it?
You can still blast it. It's when you try to copy and paste onto your sequence. So if let's say your sequence is in Word or in Primer Express, when you copy the antisense, if you will, of that probe, it won't find it on there for you because the sequence is backwards and opposite. So you just so what I do is I open up a new Primer Express window, paste it in there, tell the software to take the reverse complement of the probe and then refind it on my original sequence, and then it will show up as matching.
Another question, you mentioned you never ever changed the qPCR set and primer probe concentration, or
right.
So then theoretically you have RNA target or DNA target, you have RT-qPCR or qPCR. So theoretically then you have only 4 set of assay. And with the 4 set of kit that normally use in your lab, what if you have different qPCR machine? Would you use the same kit, same condition with the other qPCR machine, even those kit wasn't developed for that brand?
Sure. good question. So you could, but I would recommend doing like a side-by-side comparison between the two different machines or your two different kits. So for example, we used to have the 7900 from Thermo Fisher in our lab, and we had to buy new qPCR machines, and we bought the Quant Studio like you guys have. But I wanted to trust that all my assays with the same cycling conditions were going to work just as well from my quote old machine to the new one. So I was using my 7900 as my gold standard, if you will, because that's what I was using all the time. I would run the same side by side, like I'd essentially would pipette 2 PCR plates, run one on the old machine, run one on the new machine, and then evaluate my CQ values between the two of them. And once I had 100% competency that I was getting the same results, I felt confident that I can switch machines, or in what you're talking about, that you could run the same conditions on 2 different instruments and get the same results. So you would need to just do a side-by-side experiment to see if you're getting the same results in the end.
And the other question, there are so many different kits coming to the market claiming more sensitivity, robusting, and everything, and less expensive with the brand name. How would you justify to switch from one to the other to save money? Sorry.
No, it's a good question. When you're a high throughput lab, we go through like 10 bottles of master mix a month. So I totally understand, cost, right? And
Yes.
I've done this. So what I call it a master mix validation. And we do it like once every 10 years because it's challenging. And so I would ask for master mixes from different companies, like, give me your newest and greatest, or give me the one that you can give me the highest price break on, because sometimes newer master mixes, have a fancier tack in there and they can't give you a really good discount on it. So take that into consideration. I would also be a little bit leery for using not as well-known companies. Like I said, sometimes they go out of business, or their QC of their chemicals is not as good. So I would be a little bit leery from using a non-well-known company. Also, sometimes it just depends, but they cannot sometimes give you as great of a discount on their master mix. So I would personally, check with the sales rep, tell them what you're wanting, what you want to do. And you just have to do a side-by-side experiment with all the different master mixes, with all your different sample types. And if you're looking at RNA viruses and DNA viruses, so it's a very big production because you need to look at sample types, different ones, different RNA pathogens, DNA pathogens, and then the master mixes for all the different companies. So I've done this for about 6 different master mixes. I use my current one as, quote, I call it the gold standard. So I use my current one as the gold standard, and then I run all the different ones according to what they recommend in their manufacturer's guide at cycling conditions, times, ramp rates. And you just have to evaluate them. Which ones give you similar CQ values, or worse ones, or better ones? We've had ones where our negative water controls were now positive. That's really weird. So I'm definitely not going to choose that company. So I would just, you'd have to do a side-by-side comparison to really give yourself the trust that this cheaper master mix from this other company is performing at as high of a quality that you're used to.
The last question, sorry, I know people are raising hand. So have you done a spiking in your reaction to compare, or routinely use that spike as a control, internal control among the plate, among the sample, among the day performing the qPCR with the plant as a metrics?
Yeah. We do not know. There are those products that are available on the market to do that. We rely heavily on our reference gene and it being within a certain acceptable range. for calling a sample. That's what we use as our QC versus spiking something in there. So I've not used it frequently. No.
Thank you.
Fatima or Georges?
Yeah, Samantha, when you were discussing the probes, I was just wondering about the Roche UPL, you know, the Roche Universal Probe
yeah.
Library, because this used to help us a lot when you don't when we do a pileup and we couldn't find a location for a longer probe. So UPL were very short and they were, you know, readily available. I was just wondering, they have stopped using, you know, providing these probes, but is there anything in the market that you know, substitute the use of those ones?
Not that I'm aware of. That's why I did not include them in here
Because they were fantastic.
I know they were nice and short. I don't know if someone else has taken on that locked nucleic technology or not. I haven't actually looked. I just can't use it anymore to design assays with it.
Yeah. Okay. We are doing terribly on timing, by the way. So
Well, should we wrap it up and do maybe like a shorter lunch break then?
Georgios, do you want to say something before we take a lunch break?
I had a very quick question for Samantha, but I hope everybody enjoys the discussion because that's the whole idea with these workshops, so we can interact and exchange
I
ideas,
agree.
and then we can adjust our lunch break, or even leave some items for tomorrow. But my question, Samantha, is again from the other qPCR training I have attended. They were recommending to run a small experiment for every lot of a new kit you are receiving. Not only you run this manufacturer versus this manufacturer, but every time you get a new set, a new lot. Do you guys do that?
We do not do that. I do know that there are labs that do that. I think particularly like state labs or labs that have a much higher threshold for, you know, pathogen detection, like in animals that are gonna, you know, kill all the chickens out there and that type of thing. So we don't. I know some people will do that when you order like a new primer or a probe, will rerun your standard curve. I just tend to have more confidence, I think, in the lots between different master mixes. So yeah,
Okay.
so we don't. Yeah.
So Fatima, where are we in the schedule? Are we in C2 or in C3 or all the way to C4?
Let me share the badly followed agenda.
So we are 30 minutes behind. It's not that horrible.
No, we're
Yeah.
more than, yeah.
Where are we?
Well, now it's 10 minutes to 1. And we are here. I'm supposed to give an example for citrus qPCR assay design. So we're good 50 minutes off of target, off of schedule. So we can reduce the lunch a little bit.
I think we need to take the lunch break now, yeah, and then we can jump back to it. So the lunch is right now what, 15 minutes? Huh?
Can we return at 1:15, or you want to make it a little bit shorter?
Is it okay to take half an hour lunch? I mean, it is what it is. If you want to give thumbs up, and
I think we should come back at 1:15.
1:15, you said?
Yes.
Yeah, let's do 1:15 because some people are saying, you know, they're on the East Coast, so it's a little bit later back there.
Yeah.
Correct. Okay. So I will put 25 minutes on the timer.
So I was wondering what is for lunch?
You didn't arrange anything?
No, I'm kidding.
So it's Georgios, are you putting a timer?
Zoom. Yes. Thank you for
Okay.
the yeah.
So this is the portion that I'm going to show very briefly, one or two examples of a pileup and how we designed the a citrus qPCR assay. So let me share. So as Samantha was mentioning, we used to, we used to use a software called Vector NTI to pile up or align all our sequences. Now it has been discontinued. We use another software called Geniosis to pile up, and it has a portion of alignment in its application. So we take whatever pathogen is under study and we align the sequence that is available in the gene bank. Again, if we have a problem and we don't have sequences in the gene bank, but we do have some positive controls, we can create our own in-house sequences and pile it up together and create the assays at that point. This is an example of cirrhosis and CLBV. Actually, this we got a lot of sequence in the gene bank for cirrhosis, for example, and we did also create our own in-house sequences and pile it up to the gene bank location. This location was in the code protein gene. And then, for example, when we do a primer, this is the location we chose based on the criteria of the Applied Biosystems ABI software. We did in this case, I just wanted to show you that we decided to create, we start by the probe, as Samantha was mentioning. This was a region rich enough to have 10 degrees melting point, melting temperature. So we chose this location, and it also, we were very fortunate that it has a complete consensus sequences. So we designed the probe at this location. In the case of the forward primers, we had to incorporate two types of sequences in this region. One that has no, this T was not there, and another one we did it with a T. Keep in mind that this is a humongous pileup. It's we always joke with Samantha that we have our carpets ready to design our sequence from. So it's a huge length of sequence. Again, the same thing for the reverse primers. You have to incorporate the two types of probe primers sequence, one with an A, the other one without. So yeah.
Can you guys hear Fatima? I lost.
I think we lost you for a bit, Fatima.
Oh,
Fatima,
wow.
we lost you there for a second. Yeah.
Can you hear me now? No idea
Yes.
where.
Yes.
I have no idea where I stopped, but in any case, this is a consideration of designing primers, forward and reverse, in a location. You have to incorporate all kinds of sequence diversity here. So we designed a couple of primers. There's no problem of designing multiple primers. We were fortunate enough in this case to have one probe for each of our pathogens. Some of our assays also have two probes. This is another example of an assay we took from the manuscript, from applications and tweaked it out, as Samantha was mentioning. When you take the sequence and put it in the Primer Express, sometimes the temperature is not correct, sometimes the GC content is not correct, so you tweak it up to follow all the criteria, and we changed the sequence. We had one probe and one reverse. Same thing for this spiroplasma, for example. I'm just showing that we can incorporate. We have to make account of all the sequence diversity and create the primer probes accordingly. That's it. I just wanted to show you a couple of an example, and then we can go ahead and continue with Samantha's talks.
Fatima, so you guys never make degenerate primer. You physically make two set of primer with the one nucleotide difference, right?
We had, there's always recommended not to use the degenerate. I did couple of, in the 10 years ago, a probe with the degenerate primers, and I put them side by side. I designed a couple of primers and then a degenerate primer. So when you put them side by side, the idea of adding more primers was always what came the winner here. And there are a couple of publications also out there, maybe more, that always suggest that it's much better to do multiple primers as opposed to use degenerate primers. Samantha?
Yes, I'd say also generally, because when you do the degenerative primers, you don't know how many are coming with the A and how many are coming with the T. It's not going to be a 50-50 mix. So it's better to do like Fatima and I do, where we just design two primers, one with an A and one with a T in there. Yeah.
We actually had one of our assays in grapes, actually, not citrus is a much nicer commodity, by the way. Grapes is a painful, the viruses in grapes are awful. So I had some of the assays with 3 forward, 3 reverse, and 1 probe, and it really behaved very well.
So traditionally, do you go after a specific virus gene, or you look at the whole genome, or because that was clearly not the whole genome of the organism that you show us? So
no,
how do you?
We usually focus on the code protein. They usually even the conventional PCR, they focus on the code protein sometimes. And then in case of locations that we don't know which is the genomic location is good, we go ahead and and sequence a lot of sections of the genes. But most of our assays in our case are the code proteins. Gene. Samantha?
Yeah, should I go ahead and, do you want me to skip that second lecture on conventional PCR and just go to PCR setup?
I think we covered that section. I think everybody's very familiar with conventional. Yes, I think skip it and go ahead with the rest. If anybody has another questions, if not, then we can proceed with the presentations.
No, I just wanted to comment on your point, Fatima, is that keep an open mind to the audience. Code protein is the obvious target because it's highly conserved gene. But when one of our students look at the full genome of the Tatterleaf virus, actually the best sequence was a little bit after the code protein. It was part of the code protein and spill over a little bit on the untranslated region.
We designed a couple of them for Tatterleaf, you're right.
Look around on the genome. Don't just de facto go to code protein. It is the best area, but spend some time to look at the rest of the genome. Maybe you get a better sight.
Yeah. You're actually right as well, because in case of the spiroplasma, for example, the last study we had, we did had a lot of sequence of the spiroplasma, and we chose 10 different locations, and we created qPCR assays on 10 different genomic locations, and we chose two of them that behaved much better than even the code protein. And those are the ones we use for diagnostics, and those are the ones we multiplex even with HLB. You're right.
All right.
Go ahead.
Any other comments? No? And then I've already let Fatima know that she can email you, or I've emailed her all of my PowerPoint presentations, so she's welcome to either email those to you guys or share them somehow. There might be still a few spelling mistakes in there I haven't caught that I've changed, so please forgive those. But you're welcome to share those. Nothing in here is proprietary, so share them amongst your other colleagues if you need to.
Excellent, Samantha. I'll go ahead and send those in right now with all the emails that we got. If anyone in the group wants to add his email and didn't receive our invitation, our WebEx links, meaning that this means that I don't have your email. So if you would like to put it in the chat, I'd be happy to send you all of Samantha's presentation. Go ahead, Samantha.
Yeah, so this presentation is a kind of a large one, talking about PCR setup, clean areas, dirty areas, how to run curves. I'm covering a lot here. How to do your standard curve, how to make primer probe mix, quality control, genes of interest, reference genes. I'm not going to go too much in the MICE guidelines since we're short on time today, but we've already kind of talked about those a little bit. So I saw this really cool slide from Thermo Fisher talking about how the different steps of PCR introduce variants. I know this isn't really the workflow. I mean, other than it kind of shows you you're starting with your sample and ending up with your data, which I thought was attractive. But I also think here are all the steps that go into sample to qPCR to data. And I just thought it was really cool representation of all the pipetting that's involved and all the different kits, master mixes, RT, how you store things. So these are just things to think about. It's a nice combination of all the techniques that are required for good qPCR. Workflow. You're always going to start from your clean area and go to your dirty area. Okay. Your clean area is going to have your master mix in it. It can be office space. It can be someone else's lab. Maybe you don't have multiple rooms to split up your techniques in. I've talked to multiple researchers and they say, all I have is one room. How do I have, quote, a clean area and a dirty area? And all I can suggest is using different corners of your room, physical separation as much as possible. And sometimes maybe you can use the person next door's lab. Maybe they're not studying the same pathogens you are. So maybe they will allow you to pipette your master mix over there or something like that. It's a good way. You want separation and you want your clean area to always remain clean. Did you have a question, Fatima? No?
Oh, sorry, I forgot to lower my hand from earlier.
Okay. So again, what are we doing in the clean area? Okay, so your clean area is where you're going to have your master mix set up in storage, your primer probe mix. We do these in a biological safety cabinet because that's what that room came with. You could do it in a PCR hood. And most importantly, you're going to put a copy of your SOP, your standard operating procedure, in your hood. so that you know how to make that primer probe mix and you know how to make that master mix. I've touched upon this before, that your working concentrations of your primers and probes are always going to be at 100 picomoles per microliter. And that means in your final qPCR reaction, your primers are at 100 nanomole and your probe is at 80 nanomole. You're going to do your PCR plate setup in this room. And you're also that room's going to have its own set of pipetters. And they are to never leave that room, right? This is your clean room. They never go to the dirty room and then come back to the clean room. You're also never going to have samples in this room, or DNA or RNA. It's a one-way street. Okay, once that qPCR is pipetted, it never and leaves that room, it never comes back. So if you forgot to put some master mix in that plate, or you forgot to pipette something, you can take little aliquots from your clean room and take them to your dirty room, but you can never bring that PCR plate back. So, primer probe mix. I get questions a lot on how to make this, okay? So I've divided the SOP into 2 pages so that it fits on here, but this is the first part. And the first line is that all primers and probes are to be at 100 picomoles per microliter concentration. That's your stock solution, and that's what's being stored in your freezer. So you always can assume, which generally in science you don't like to make that any assumption, but you're always going to assume that your stock concentrations are at 100 picomoles per microliter. And when you're making your primer probe mix, you're going to be mixing 20 microliters of your forward, your reverse, 4 microliters of probe, and 196 microliters of water. The important part here is this 240 microliters final volume. So in that previous talk, which Fatima was giving, that we had to design 2 forward primers and 2 reverse primers on that one pathogen, you would reduce this by 40 microliters, because now you're having 20 microliters of forward 1, 20 microliters of forward 2, 20 microliters of forward reverse 1, and 20 microliters of reverse 2. You're not going to have those and make it equal 20. So you're not going to add 10 microliters of forward 1 and 10 microliters of forward 2. You're not going to do that. So you just adjust how much water you add to your primer probe mix, because you want that final volume of 240 microliters so that you have your PCR concentration and your master mix to equal what I showed you in that previous slide, to 400 nanomolars and 80 nanomolars. So sometimes it's a little confusing on how to get to that 100 picomoles per microliter. So here's an example of what the sheet, when you order primers, gives you the sequence. It gives you how many nanomoles are on the tube. That number is pretty much always on the tube on part of the label. GC content, TM, if you notice, these will be very different than what our software used, the Vector NTI, Primer Express, because again, different softwares calculate this differently. And then they're always really cool, and they tell you how much water to add, okay? 100 picomoles per microliter is the same concentration as 100 nanomole. So if you have a tube and it shows that you have 14.9 nanomoles in there, and you don't know how much water to add, move the decimal place to the right one spot, and that's how much water you would add. So in this example, you would add 149 microliters of water, okay? So also in this clean room, we're making our master mix. Again, our lab is all about the cheat sheets, because I don't want to have to think too hard about how much concentration or how much volume to add of everything. And I want everybody to be doing it the same exact way. So if your lab uses 3 different master mixes, you have a singleplex, you have a multiplex, and you have a one-step, you would have 3 different SOPs on how to make those master mixes. Okay, this one, we have it divided by groups of 10. So if I need to make 30 reactions of this mix, I'm just going to follow these volumes right here. And I know that's going to be enough for me to do 30 PCR reactions. I would say if you really have to do 30 PCR reactions, you need to add a little, pipetting error or 10% extra. So I'd probably make a little bit more than this. But have these cheat sheets in your hood, easy to use so everybody can follow them. This is what our hood looks like. So here's our SOP on primer probes. And then we have 2 different types of primers and probes. So here's our master mix list on how to mix both of these. Here's all of our pipetters. They live in this hood. They never leave. Here's just a diagram from a company, IDEX. They run a lot of animal PCR. And this just kind of shows, they have their clean room, their neutral room where they're doing their PCR reactions, their sample prep room. And then when I did get to visit this place, they had this cool little like sliding half door here where they transferred their PCR plate. So it didn't even come out of this room. I mean, obviously this complex was designed specifically for PCR. You're not going to have that generally in most labs. So your dirty area.
Samantha,
Yes.
On your pipette earlier that you said they live there, never leave, and so on. How often do you calibrate your pipettes?
Once a year.
Okay.
Yep. Your dirty areas, okay? Depending on what you're doing, you can have multiple dirty areas. We only have one room because we're not culturing anything. If you're culturing something, that's going to need to be in a definitely separate from your extraction area or from your clean room. Our dirty area has our biological safety cabinet, which is where we open up our samples and put them into the extraction plate. This is where you're going to be pipetting your DNA or cDNA onto your PCR plate. We do that in a PCR workstation because we just want to keep it clean from the environment. Also, if you're doing a lot with plasmids, I mentioned liquid cultures already, I would even get a second room. So we have a shared room that's on the opposite side of our building where we open up our plasmid DNA. So we don't even do that in our dirty area because we're so worried about contamination of that. We open that in a separate lab. In our dirty area, each section has its own set of pipetters. So we have a set of pipetters where our sample prep is, another set where we're adding our DNA and RNA. So you're going to need a minimum of 3 sets of pipetters, one in the clean area, and then one in your sample prep area, and one in your PCR area. I would say you're probably going to need at least a P10, P100, and P1000. I think last time, we had our pipetters calibrated in December, and I think we're close to 70 or 80 pipetters between the 2 rooms. If you're really getting into qPCR, you're also going to need electronic pipetters to do all the aliquoting for you. So we have a whole set of single ones and 12 well ones for when we add our DNA or RNA to our PCR plate. So you're going to need, and those pipetters are very expensive, over $1,000, but you need to invest in those to be able to increase your productivity, excuse me. Also, don't forget, you're going to want filtered tips, and you're going to want to use filtered tips all the time, again, to prevent contamination, aerosolization of a positive sample just into the PCR shaft is where you're going to get contamination there. So only filtered tips. We also only use tips that come from the company of that pipetter. So we only use Rainin pipetters in our lab. In my opinion, they're one of the best. And we only use Rainin pipette tips. We don't use any of the knockoffs. I've not found them to be as good as Rainin. Particularly, you'll see that on the 12-well pipetters, that you'll get varying volumes when you go to pick up a liquid. So moving on to standard curves, we've talked about how each assay has to have a standard curve run on it. I'm have already touched a little bit about this, so I'm not going to go into too much detail, other than you really need that slope and y-intercept to get your calculations. If you're doing a 10-fold standard curve, you should expect a 3.3 CQ difference between each point. So you can kind of always do a quick little calculation in your head. If someone's talking about 2 CQ values and they're 3 CQ values apart, you can always just say, well, there's about a 10-fold difference in the concentration of that pathogen between those two samples. 5-fold curve, there's 2.2, and a 1-fold is just about a CQ of 1. I mentioned this, I think George asked questions about tips for standard curves. Like I said, I use the same pipette tip, vortex it, heat it. These are just all little tricks that you can try to use if you want to. So here's your standard curve. Again, I have my low value down here, my 36. It'd be ideal to have one more up here where I would assume it would be at 40. I also do the average standard deviation, the differences between these 2 points, because you expect it to be 3.3. And then you graph it. Okay, so here are the equations. I didn't have that on that previous slide for you guys. So your efficiency, it gets plotted into here. Your y-intercept helps determine your sensitivity. You generally want your y-intercept right around 37 to 40, okay? That's going to give you really nice sensitivity and detection of low copy numbers. Master mixes. We kind of also had questions about how do you choose a master mix? How do you, when do you stay with it? There's all different types of master mixes out there. And if you do, if price is a big concern of yours, work with your sales rep, tell them these things, because sometimes they might, their best seller might be their most expensive one, but the one they can give you the greatest discount is an older one. Our master mix that we use, the universal master mix, was, put together like 15 years ago. It's old in the standard of master mixes, but whenever we do a comparison, it's always one of the best. So I'm not always convinced that some of these new ones are all that great. Don't forget, when you're working with your master mix, you want to make sure it works with all your sample types or all your pathogens. Most of them nowadays come with the hot start. Be careful of bubbles. Also, most of them, except for when you're pipetting RNA, the master mix itself is stable at room temperature, generally with up to 48 hours. Yeah, sorry.
Sorry. You mentioned you do once a year calibration. May I ask how much workload you have per day?
Oh.
Because the workload define how often you should do calibration. If the lab is getting 100,000 per month, then maybe in 3 months you should have calibration. So
Sure. Yeah, we process about 100 samples a day would be about our average workload.
thank you.
Yeah. Also, they do, I don't know if it's in this talk or another talk, there's some kind of nifty, let me see, in the lab instruments that you can test if your pipetter is pipetting accurately. It's not in this talk, it must be in tomorrow. Rainin sells this little box, and you set your pipetter, let's say to 10 microliters. You pipette your 10 microliters in there, and it says, if it passes or not. I guess, like if it's in one or like within half of a microliter type of thing. So there are in lab devices to check your pipetters if you're really concerned about that.
I'm sorry, follow-up question about another. So when was the longest time you left your master mix without adding target on the room temperature, prepare it in the morning, and then add your RNA and running it, and still you get the good result? When you said 48 hours, master mix would survive in room temperature. Have you tried it?
Heck no, are you crazy? No, I'm just teasing. So our lab, we keep everything in the refrigerator like during our work day. So if we pipette a PCR plate in the morning, even if it does include target or if it does not include, either way, target, we keep it in the refrigerator until that afternoon. We do have a robotic twister on our Quant Studio 7 that runs plates overnight because we have so many. So I've set plates that's with my master mix. Granted, this is cDNA, and my target in there, and they have sat mixed together at room temperature for up to 12 hours. And my QCs look great. And the master mixes, a lot of them on the companies promote that their master mix with your target is stable for up to 48 hours. I've never personally gone that long. I kind of like to mix it and run it as soon as possible. But we do run plates overnight all the time, and QCs look just fine.
So with the robotic feeder, arm robotic feeder, you don't have any problem. So you can run the Quantum Studio overnight, like a full-time shift.
Yep.
Okay.
Yes.
Thank you.
Yep.
I have a question for Samantha.
Yeah. Yes.
How do you approach a situation in which your gold standard master mix, for example, is discontinued or is being improved upon?
Yeah, I know one of your QuantiFast kits is being discontinued. I would try to get my hands on some of the current one, which I would at this point call it your gold standard. And then I would test 2 or 3 other ones, probably what the company is recommending as a replacement, and maybe other ones from other companies. And you're just going to need to do a side-by-side experiment. Like I said, run a whole bunch of different sample types and a whole bunch of different viruses or bacteria with your gold standard and with your new ones, and evaluate which ones are giving you like either better CQ values or the same CQ values.
Thank you.
Mm-hmm. So this touches right upon that point you just brought up. So when we and you guys, Riverside, are doing singleplex or multiplex with DNA or cDNA, we're using the universal master mix, which is all we use in our lab because we do cDNA all the time and we don't do multiplexing. There's the gene expression by Thermo Fisher. Singleplex, one step, is the AgPath. And then this one right here, this the QuantiFast by QIAGEN is being discontinued. I was still able to order some for our side-by-side experiment that we're going to do. And it's recommending the QuantiNova as its replacement. But you guys really need to do a side-by-side to make sure that master mix works well. There's fast master mixes out there. I have not been impressed by them. The selling point of PCR in 40 minutes sounds terrific instead of hour and a half. But I have lost at least a 10-fold sensitivity in those. So I've been super disappointed. And then don't forget there's master mixes if you're doing SNP or high resolution melt or digital PCR. So the different techniques are going to need different master mixes. This is just what I was saying, the fast master mix. You do need a newer PCR instrument because you need a faster ramp rate. So you can't do this on the older instruments. Do a side-by-side comparison. You're going to have to test it. You can't just trust what the sales rep or what you're seeing slides they're showing of how great it is. How does it work in your hands? Because I've yet to be impressed with any of the new master mixes on the market. Snips, I'm always just find it so cool that it can tell, a G from a C, and it calls a heterogeneous. I just think it's really cool. I have one virus and one bacteria where I do SNP determination. And you can just get this really cool graph at the end, and you just know right away which sample type is which. I'm just going to touch about, just to give you some other terminology. I've never personally done high resolution melts. It's kind of like cyber, where you get a dissociation curve, and depending on if you have an A, T, G, or C, you get these curves at different locations. You do need a special software for this. You can run it on the Quant Studio, but you just need special software. Digital PCR, I've never done this either. It's supposed to be a really cool, quick way to get an absolute quantification number on a sample without having to run a curve. It's just the way you load this chip. Each little well is supposed to get one sample. If it's positive, it's supposed to give you how many target molecules you have. So there are just other techniques out there. Maybe it doesn't fit what you're doing right now in the lab, but maybe it'll fit with, something in the future. Generally, most master mixes have rocks in there. That way you can know that your PCR conditions are nice and stable within that well. So it's a passive reference dye that should remain constant throughout all the cycling conditions. It should not change. Nothing should affect that dye. Also, a lot of master mixes have UNG in them. And what this does is it uses a DUTP instead of a DTTP. And that way it helps prevent lab contamination if you have to open up that qPCR plate, which hopefully in reality you never have to do, because now there's U's in there instead of T's, and you shouldn't get amplification with another set of primers. Also something, to think about is, why are you only running 40 cycles of PCR? Isn't more better? Why aren't you running 55 cycles of PCR? Because I'll see that in manuscripts where they ran 45 cycles or 50 cycles. I'm just like, wow, that takes so much time. And that's kind of it. So there's no really good reason to run more cycles, because when you do absolute quantification, your detection limit of one copy is around 39, a CQ of 39. So running a cycles of 50, that's not going to change. You also run chances of higher primer dimer or non-specific binding of your primer. So I would never recommend going above 40 cycles. Some people even only do 35. Some people believe a positive CQ value between 35 to 40 is a false positive. I don't believe that, but there'll be people out there that'll tell you that. Again, put your SOPs in the hood. Make it easy. Don't allow for any confusion between your staff of how to pipette anything. I just can't stress this enough. When I first started, I saw all these in the hood, but no one told me the difference between this one and that one. No one explained it to me. So when I was pipetting my qPCR plate, I guess I was young, dumb, embarrassed to ask questions. I don't know. I would ask that question nowadays, but some days I chose this one and some days I chose that one. So make sure you explain everything to your staff or your fellow researchers, especially new people coming in the lab. They should not be left alone in this hood until they know what all those SOPs are. Sample quality control. Okay. When I get a new project, or like, let's say for you guys, a new tree type or citrus type, and you want to know where these viruses are found in there, you're going to want to test several different areas, right? So I would suggest 6 different samples. You're going to want to test the background if you're doing 2-step, and you're going to want to see what the reference gene is. Is it the same between the different areas or different treatments, or is it different? Okay. Also, one of the things you're going to want to set in your lab is a protocol for a failed quality control of a sample. So what value of what CQ value do you use in your facility to say, hey, this sample failed? We either need to re-extract it or rerun the PCR, or it's passed and I trust the negative result is negative, or I trust my positive result. So we have 2 different set points in our lab. So one is called GAP-DH, and its CQ value has to be under 32 to be considered passing. So if I run a sample and my GAP-DH is at 33, I do not release those results, okay? We have another reference gene called eukaryotic 18S. Its cutoff is 30. So again, if a sample has a CQ of 31, it's considered failed quality control. In our facility, we will test each sample 1 additional time for free. So if the sample has an 18S of 32, I will re-extract that sample and I will rerun it for all those pathogens free of charge to my client. That's what we offer. Some people will just report that that sample has failed quality control and there is no additional testing. Yeah, Sora.
So you run for every single sample one of these control or both control, and is it one or triplet? How would you do it?
We run one or the other. It depends on the sample type, which one it gets. And we only run it in single.
And for every single sample you get?
Every single sample.
Thank you.
Because we run it on every single sample because this, to me, the reference gene is kind of my extraction control, if you will. Also, it tells me, if there's potentially inhibitors in the sample, if it fails quality control. Sometimes if your elution has a little bit of color to it, more particularly for us, blood samples will elute a little dark because of the hemoglobin in there, they will fail quality control. So yes, every sample always has a reference gene.
Sorry.
If it fails, yeah.
And if you run it one time and then you are running several tests or several pathogen on the that sample in different days, would you run it again or one time satisfy your quality control?
I run it every time.
Oh, okay. Thank you.
Especially, yeah, I mean, if there's been a freeze thaw or you have to make a new dilution, but yeah, it's our protocol in the lab. We run it anytime we run into qPCR for genes, it always gets a reference gene with it.
Thank you.
Sometimes if you don't want to re-extract your sample, maybe you don't have any more of that lysate, you can dilute your DNA or RNA. Again, it probably have some PCR inhibitors in there. Sometimes if you overloaded your column, your lysate might be kind of too much material in there. So you might dilute your sample before you extract it. That'll also help kind of dilute out some inhibitors if that's the issue, or just dilute out, like you have too many cells in there, it'll cause inhibition later on, which is why I talk about not overloading the column. Look at the brochure from the company. It'll tell you, hey, this column can handle 1 million cells. So don't put 10 million on there. It can't handle it. So if your sample is very strong, you might need to do a 1 to 10 dilution of your lysate so that you don't overload that column and that you can get proper extraction and elution. Yeah, sorry.
So you mentioned in your master mix, you take 7 microliter per reaction. So theoretically, you put 5 microliter of your target. So the procedure you use, MagMax or column, it give you relatively good quality RNA and DNA. Would it be
Yes.
too much to add 5 microliter per reaction?
Yes, it would be.
So then how would you actually incorporate? So whatever you get from MagMax, are you going to dilute it or add
Yes.
more? Okay. So you
No, I'm gonna dilute my nucleic acid.
each time, or you do it from the plate, you make duplicate plate with the dilution, you use that one for the subsequent test, and you keep the original one freeze.
Yes,
Okay.
yes, just what you said.
And would you consider RNA DNA target with a higher volume, or you bring everything the same level? For instance, that target, if you want to run is TNA, if you want to run DNA target, you still use 1 microliter of diluted one. For RNA, still you're using 1 microliter for target one.
Yeah, it's a good question. So I'm trying to think what we do. So DNA, we do 1 microliter as our standard, like, first step. I think for our seed, since we make cDNA, I think honestly, I think it's about a 1 to 20 is what's being run in the final PCR reaction. By the time we do the RT and add a little bit of water to it. Now, as I said, we usually use 1 microliter of DNA as our normal default in the lab. There's exceptions to those rules, obviously. So when we process fecal material, which we know has a lot of inhibitors, that default dilution is a 1 to 20. So you might need to set different dilution factors for different sample types in your lab.
So then become complicated. As a diagnostic lab, if you want to practice the simple way, then for tighter consent calculation, I'm going to make all of these problem later on. What was the dilution? So I'm calculating it correctly or not? So it become more complicated, right?
What are you using the calculation for? I missed that part.
No, if you want to go pathogen, how many particle you have, how many viral load you have, then by dilution, all of these calculation become a little bottleneck for a smooth processing.
Yes and no. I think it's going to depend on how you're going to express that absolute quantification. If you're going to express it, let's say how we do it in our lab is per million cells based on my reference gene. So my reference gene has been diluted the same amount as my target gene, right? Because it's the same DNA, just in 2 different wells. So when I don't need to worry about any dilution factor, if you will, when I'm doing absolute quantification, because I'm basing it off of a number of cells. I would agree with you if you're going to say I have X amount of pathogen, let's say per microliter of DNA or per gram of tissue, then yes, your dilution factor will, you'll need to take that into consideration when you do that calculation.
Okay, so for the gap DH, how many copy we have for that target and for 18S per cell?
Yeah.
You know?
So gap DH only has 2, which is one of the reasons why we use that one when we're doing viral or bacterial loads. We don't ever use 18S because there's multiple copies of that per cell. So you need to, if possible, you want to choose a reference gene with a known number of copies because you will need to take that into consideration during that calculation. So we only use GAP-DH when we're doing viral or bacterial loads because we know there's 2 copies.
Thank you. Have you ever reduced your volume, reaction volume to half in 96 format, still getting good result, or when you do 384, that volume would be significant, good enough for reaction? In a soft 12, have you run 6?
I've run 6 and I run 5 in a 384 well plate.
But not 96.
No,
Thank you.
we only use 384 in our lab. We don't have any 96 well blocks or anything.
So if we are running 96 format, would you recommend us to switch to 384 plate? And
Absolutely.
would you please give us the pro and cons? Sorry.
Yeah, I think one of the main pros is going to be you're going to be saving on half of your master mix immediately, right? Because how many? What is your PCR volume in your 96 well plate?
12 usually.
Oh, you do 12. Okay. For me, I can just get that many more. So let me backtrack. So like I said, we get about 100 samples a day. And on the average sample, I'm running 10 different pathogens. So for me to be able to fit all those samples and all those pathogens on, I need 384 well plates to fit that all in. So for me, it's just straight like math to be able to get high throughput. That's one of the main reasons we only use those plates in our lab. I don't normally do a 5 microliter reaction. It's only kind of on a special case type of thing. We always do 12 microliters.
Even in 384.
Yeah.
And then how would you operate that one? Are you always using robotic for this, pipetting? Or you
No.
actually...
I'm the robot, and so is my coworkers.
Okay.
It's all done by hand.
Okay. Thank you.
Yeah, there are robots out there that can pipette that master mix in there for you if you're always testing the same pathogens. And if you are, I would totally recommend that. Ours varies all the time. So sometimes I'll have 10 respiratory panels and 10 GI panels and 10 blood panels. So ours changes day to day. But if you have a known set project where I'm going to test for these same 10 viruses, I'm going to test for them, on these next 10,000 samples, yes, I might look into a liquid handling station that will pipette that master mix for me. And they do exist, and they are very good.
And since you mentioned every plate is different, do you have the master template for whatever pathogen you are doing to use that one over and over, or each time you do it yourself?
We do it ourselves each time.
Thank you.
Let's see. Sample quality. Generally, you're going to have a higher sample quality from either a very, very fresh sample. If that's not possible, you want to freeze it. Prompt freezing is going to give you the best results in the end. Sometimes if your sample, not quality, but quantity is very low, sometimes you can try to concentrate your DNA or RNA. There are columns out there, or you can also do it by hand. Talking about sample quality, if you want to nano drop your sample, so it's this cute little machine right here. What's really awesome is that it only needs 1 microliter of your sample on there. It gives you, your 260, your 260, 280, your 260, 230. So right away about that. We don't use this too much in our lab, but we do have it sometimes. Another really cool sample quality check is the Agilent bioanalyzer. This is needed for next generation sequencing because the quality for next generation sequencing needs to be fabulous for it to work. It needs an RNA integrity number or a REN number of greater than 7 before you can do sequencing with that. And what's also really neat about the bioanalyzer is it quantifies it and So you get quality of it and you get an amount of it. So this is what a really pretty one looks like. You get nice 2 peaks. And this is an RNA sample that has degradation. These can be a little bit trickier to run. The chips are a little particular sometimes, but it's a nice tool that we have in the lab for when we need to really know how the RNA is looking. Some people have this qubit in the lab, and it uses fluorescence to measure your DNA and RNA. I've never used it for this. I have it for another technique. So you might have this instead of the nano drop. One thing to also think about for qPCR is you don't need the best concentration or quality of sample to get good qPCR results. Because if you think about it, our amplicons are so small, right, of 150 or less base pairs, that it's kind of okay if the DNA or RNA is degraded a little bit, because those primers and probes can still bind in there. If you are running RNA, you generally do want it free of DNA. So some columns will do an on-column DNA digest. If you have inhibitors in there, you can, try to purify your sample. There's column purification methods. You can also precipitate your RNA and try to remove any contaminants that way too. I try not to do those things because you lose information. You lose quantity every time you try to clean up your PCR. Someone earlier had asked about kind of spiking a known exogenous source into your reaction. So Thermo Fisher has this xenosynthetic RNA that you can spike into your sample total nucleic acid and check your reverse transcription and your PCR. Like I said, I've never used it, but it's available out there. I thought this was a cool little slide again from IDEX, which is a private company, showing like what each of the controls are doing for you. PCR control is looking for contamination, or a positive is looking for function, making sure your master mix is made right. So I just thought this was kind of a cool representation of what all the controls are doing for you and why you should be running them all. So reference genes, it doesn't sound like this applies too much for you guys since you're looking at viruses, but if you're doing genes of interest and you're trying to figure out up or down regulation, you can run, one gene of interest, or you can run a whole plate of 96, it's all over the place. Something to consider even with pathogen detection, are you running everything in single kit or are you running it in triplicate? The MICE guidelines suggest triplicate, but that can become cost prohibitive depending on what you're doing. We've already talked about exon-X injunctions, excluding genomic DNA contamination. Talking about reference genes, yes, you need one for every single sample. This is how we in our lab determine if we believe the negative results for the pathogen or if we don't believe them. You got to have a cutoff of your reference gene to know if your sample passes or fails quality control. What if your gene of interest or even your virus amount, what if it's really low? What if when you're running your qPCR, this virus normally comes up, let's say around 39, really, really weak, and you kind of question, like, is it really there? There is a technique where you precipitate your sample, your nucleic acid, use ethanol, salts, and glycogen, and you pellet your nucleic acid, and then you do what we call preamplification. Essentially, it's conventional PCR, but with the same qPCR primers, no probe, just the 2 primers. So you're kind of doing a conventional PCR reaction first, and then you take that product, and then you run your qPCR. and you'll see a sample go from a 39 to like a 26. So it's a technique we use very sparingly, where you're pre-amplifying or really boosting up that signal for different viruses. We'll do that sometimes when we think the viruses are kind of hidden in different tissue types and we're not sure if they're there. We'll pre-amplify them to try to find it. Sometimes you'll need to do a reference gene validation and make sure that reference gene is detected in all the different sample types, that it's constantly transcribed between your different sample types. You don't want a reference gene that expresses differently in different parts of the plant material. You want it to always be the same. The perfect one does not exist, but you can, test different ones and try to choose one that works for your facility. Talking about reference genes, you always want to pipette that one last in your PCR plate. So here is a panel. These are all different viruses. This is the bacteria found within the horse. Here's GAPDH as my last target. So when I pipette my DNA or RNA, I'm going to start here at the top and do this as use the aliquot on your multi-channel, and it's going to come here, and I'm just going to use the same tip and just do 5 little aliquots all the way down to the bottom. You always want your reference gene at the bottom because it theoretically, right, should be the strongest CQ value. And if you had GAPDH up here, and if your pipette tip became contaminated, you might bring a false positive into your next well. So you're always going to want that at the end. Also, it's a little confusing, but when you're coming from, so we store our DNA and cDNA in a 96 well plate, yet our PCR reactions are in a 384 well plate. When I come from the 96 well plate to here, it goes into every other column. So it's a little confusing when you first move to the 384 well plate. But you can just run so many more samples at one time, which is what we need to do as the high throughput lab. I skipped this. This was the previous talk about how to do absolute quantification, so I'm not going to go over that right now. We've kind of touched bases a little bit about the MICE guidelines, the minimum information for the publication of quantitative real-time PCR experimental guidelines. Several years ago, people were getting very frustrated about what little information was being published about qPCR, like where primers weren't being published, efficiency wasn't out there. When people were reproducing results found in publications, I thought this was really interesting. They found about 30% time that negative controls were really positive when another lab did them. So it's something to, so some gentlemen got together, produced these guidelines to encourage people to publish correct information. There's the paper and the citation for it. What's covered in here? It's very extensive. They went to a lot of work in there. They talk about sample prep, controls, target gene information, assay information, specificity, efficiency, sensitivity, which chemistry was used. Did you do reverse transcription? What method did you use? How did you analyze your data? It's, like I said, it's a very extensive publication. And also what I thought was interesting is that every fact that they think you should publish, they tell you if they should be essential or desirable. So in the MICE guidelines, it says it's essential that you publish your primer sequence, but it's desirable for you to publish your probe sequence. And this list, I think it probably has maybe 100 different facts or stuff like this that they want you, that they have listed as essential or desirable. according to this guideline. So the list just goes on and on. I kind of just put the most common ones just to give you some examples of what's in this guideline. And that's all I got for this one. This was just kind of a catch-all, general, how to set up your lab, how to set up your workflow, things to think about. So are there any questions?
I have a comment, Samantha.
Yeah.
The classic Mikey from Bastin et al., the 2009 one and the most updated one, they are focusing on, that guy. They're focusing on quantitative PCR, gene expression. I found another paper, and maybe Fatima can bring it up, what they call Mikey for qualitative PCR. So they follow this Mikey, but they based it on a problem they had to solve with transgenic plants, and they wanted to see if the transgene was there and how many times it was there, how many copies of the gene that they were introducing to an organism was there. So it was a yes or no versus how much. And I found that paper, the qualitative PCR Mikey, much more applicable and useful for the diagnostics labs.
Oh, cool.
I think
Okay, yeah, I'll take a good look at that then.
Yeah, Broders et al. I will find it, put on the chat, or if Sorab or Fatima are faster than me. Broders et al. 2014.
Yeah, I have it sorry, I have it in my talk. I will just put it in the chat.
Yeah. So for all of you, simplified, because we don't do the quantitation part. I mean, you can always do quantitation in how much pathogen you have, but for us, it's typically yes or no.
Yeah, I think most answers for diagnostics, it's yes or no, because even if you give an absolute quantification number, like, what does it mean, right? Are you infectious at 10 copies, or are you more infectious at 1,000 copies? But, you have to do so many experiments to determine that, COVID's not even expressed, with an absolute quantification. So I think, yes or no, positive or negative is sufficient for most diagnostics. Are there any other comments or questions?
Fatima, if you have also thanks for putting it in the chat. If you have the journal, and if you have a DOI or something, it would be, but this is enough.
I'll get it, definitely. Have the
Should be enough for everybody to find, but capture it, guys. All of you in the diagnostics labs, at least for me, made much more sense, this paper. We use both and we reference both on our papers, but that guide made much more sense because, again, is the transgene there, yes or no? Which is exactly the question we're asking. Is the pathogen there, meaning in the plant nucleic acid soup, yes or no?
So if no one has any other questions, I did send an email with all the presentations to all of you, even the people that I did not have their emails, and they just placed their emails on the chat, including also the talks for tomorrow. So if there's anyone who has any comments, if not, then we will adjourn and meet again at 9 a.m. tomorrow.
Will the recording be available with the talks you just sent over?
Yep. Irene, right? It will be available. I mean, we can send it again after the 2 days. We'll send all the talks as well.
Okay, great. Thank
The
you.
recordings, yeah, Jonathan, I see speaking. The recordings, we will double check whatever rules there are, you know, just in case there is something that needs to be trimmed out, or we will double check with the institutions and get consensus from the speakers and so on. And we said we want to record this for training purposes. So yeah, we should make it available. And I don't know if we want it on a website or just to share it with the participants. That's another decision another discussion we can have. Maybe just for the participants, I don't know. Okay. Thank you all for joining. If nobody else has a question or a comment, I enjoy the presentations and as something as important as PCR and qPCR for our labs is very, very critical to start digging on some of the basic ideas, so we can all speak the same language. And that's how we saw it in our team. No matter if you were the computer tech guy or an administrative assistant, or you were working in the lab or in the greenhouse, we thought that if everybody listens this, then they can improve the work, they can improve the quality of the work, because now they understand what it means. It has to go in the lab for PCR. What does that mean? So the person works in a greenhouse now, knows what it means and how they need to collect a sample and what to do with it, how to store it, how to transfer it, and so on. Not only the people in the lab. And the same way with our admin assistant, when they share results with our stakeholders, they know what is going on behind all this, and they can use different language and explain to stakeholders about the results and about the process and the timeline and so on. I see similar comments in the chat. So again, thank you. There's no reason not to finish earlier.
Nope.
Especially for those on the East Coast. And we'll see you tomorrow. Anything closing remarks for you, Fatima and Irene?
I'll go ahead and thank everybody for participating. We look forward to seeing you tomorrow. Thank you so much, Samantha, for all the talks. And thank you all.
You're welcome.
Okay.
Yeah, this was great, Samantha. Thank you very much. The only thing that I would like to add, we might prepare certain questions for you guys to answer, like kind of comments or assessment of the training. As we mentioned, that this is kind of a guinea pig trial for a larger scale training on qPCR. So we'll just send some points by email, and we'll be very grateful if you can just provide us with any feedback. That will be very helpful for us. And thank you again, Samantha.
Yeah, you're welcome. I'm glad to hear all the discussions after the talks. I think that's, to me, also interesting, where people are bringing in their own personal experiences and what's working for them and what's not working for them. And this is a great time and a safe place to talk about that, you know? I think that's important. All righty then. We'll see you all tomorrow.
Bye. Yeah.
Bye.
Bye.
Thank you.
Good day.
Bye, Samantha. Thank you.
Thank
All right,
you.
bye.
Bye. Bye-bye.